In a laboratory at the University of Rochester, on a night in the early years of this decade, a small brain was lit from within. The mouse was alive. Its skull had been opened with care, a thin window of glass sealed over the cortex and into the cerebrospinal fluid that bathes the brain's outer surface. A fluorescent tracer had been introduced, a molecule small enough to drift with fluid currents, bright enough to be tracked by the instruments above. The room was dark except for the cold blue glow of the microscope. The researchers were still. And then, in the imaging field, something moved. The tracer did not drift randomly. It did not spread in all directions the way a drop of dye spreads in still water. It moved along a path, a specific, repeating, arterially defined path, entering the brain tissue through the narrow channels that sheath each penetrating artery as it dives from the surface into the deeper tissue below. The movement was slow. It was organized. It had been going on, unobserved, for as long as there have been sleeping mammals, and no one had seen it until then. What moved that night in the blue-lit quiet of that room was cerebrospinal fluid, and what the fluid was doing, what it had always been doing, was cleaning the brain. The brain had a drain. It had always had a drain, and the drain only ran at night. Macon Nedergaard had spent years thinking about the spaces between cells. Her laboratory at Rochester had been working on the biology of glial cells, the non-neuronal cells that make up roughly half of all cells in the brain, long considered secondary to neurons, long described as merely structural, scaffolding and support. She was not satisfied with that description. Glial cells were not passive. They were responsive, chemically active, spatially organized in ways that suggested a function more precise than simply holding neurons in place. The question was what that function was, and whether it connected to something the brain needed to accomplish at a scale the field had not yet thought to measure. The answer came through fluid. It came through a question so basic it had been mostly overlooked. Where does the metabolic waste of the brain go? Every tissue in the body generates waste. The liver produces it. The muscles produce it. The heart produces it. The body's solution, worked out over hundreds of millions of years of vertebrate evolution, is the lymphatic system, a second circulatory network running alongside the blood vessels. Its vessels collecting the interstitial fluid that leaks from capillaries into the spaces between cells, picking up proteins and cellular debris and carrying them toward lymph nodes where immune cells filter the load before the fluid returns to the bloodstream. The lymphatic system is everywhere. It drains the skin, the gut, the lungs, the muscle. It is the reason a swollen gland in the neck is a sign of infection. The node downstream of the throat's lymphatic drainage is working harder than usual. But the brain has no lymphatic vessels. Open the skull, trace the anatomy with the most careful instruments available, and no lymphatic network appears. The brain, the most metabolically active organ in the body, consuming a fifth of all the energy the body burns, despite being 1/50th of its weight, generating enormous volumes of molecular waste with every second of operation. This organ has no lymphatic vessels at all. Hold that image briefly. Picture the body's lymphatic network as it is drawn in an anatomy textbook, the branching vessels collecting fluid from the muscle and organ tissue of the arm, the leg, the chest, converging toward the lymph nodes clustered at the groin, the armpit, the neck, the fluid moving slowly inward through a system of one-way valves and smooth muscle contractions. The whole network, a quiet parallel river running beneath the skin and through the organs. Picture it mapped and complete and running throughout the body, and then notice the one place it does not go, the skull. The brain inside, the blood-brain barrier is part of the answer. The brain's capillaries are sealed differently from capillaries elsewhere in the body. The cells lining them are bound tightly together, their junctions close and selective, letting through only what has been specifically cleared for entry. Immune cells patrol everywhere else in the body, circulating freely through tissues, surveilling and responding, and the lymphatic system carries their traffic. But the brain keeps immune surveillance at a distance. The same barrier that protects the brain from pathogens and toxins in the bloodstream also makes it incompatible with the conventional lymphatic architecture the rest of the body uses. And the skull itself constrains the solution. The brain is enclosed within a rigid fixed volume box of bone. The volume inside cannot change. Any functional system operating inside that space must account for the pressure dynamics of a sealed container. Fluid added in one place must be displaced somewhere else, and any network of vessels must be integrated into a space with no room to spare. A conventional lymphatic network with its own vessel walls and its own structural demands cannot be added to the interior of the brain without displacing something that is already there and already essential. So evolution arrived at something else. The solution is built from cells that were already present, already surrounding every blood vessel in the brain, already in contact with the fluid spaces that thread through the tissue. The solution is built from glial cells, specifically from a type of glial cell called an astrocyte, and from a property of astrocyte membranes that makes fluid transport possible without any dedicated vessel infrastructure at all. Astrocytes are among the most numerous cells in the brain. They are star-shaped, their many projecting arms reaching outward in all directions through the tissue, making contact with neurons at synapses, with other glial cells, and most relevant here, with blood vessels. Every blood vessel in the brain, from the large arteries on its surface to the finest capillaries threading through the deepest tissue, is wrapped in a sheath of astrocyte end feet. The end foot is the terminal structure of one of the astrocyte's projecting arms, flattened and pressed close against the vessel wall like a tile of living membrane, its inner face toward the vessel and its outer face toward the brain tissue beyond. In cross-section, the kind of image made visible by electron microscopy, the technique that resolves structures at the scale of molecules, the vessel and its astrocyte sleeve look like a pipe wrapped in a close-fitting cuff. The end-foot tiles press so tightly against the outer wall of the vessel that the gap between them is barely measurable. They do not fuse with the vessel. Between the end-foot layer and the vessel wall, a narrow fluid-filled space remains, the periarterial space, sometimes called the Virchow-Robin space, and it is through this space that cerebrospinal fluid enters the brain tissue. The cerebrospinal fluid itself is produced continuously, day and night without pause, by a structure buried deep in the brain's interior called the choroid plexus. Picture a cauliflower, dense, folded, its surface area multiplied many times by the intricate layering of its form, and then picture that form rendered in living tissue, hanging within the fluid-filled chambers at the center of the brain. The choroid plexus is exactly that, a folded vascular secretory tissue, whose business is the production of cerebrospinal fluid, drop by steady drop, every hour of every day, for the full span of a life. In a human brain, the choroid plexus produces roughly half a liter of cerebrospinal fluid each day, a volume that fills the brain's internal ventricles and the subarachnoid space around the brain's outer surface, and is then continuously absorbed and replaced throughout every waking and sleeping hour. The fluid the choroid plexus makes is clear. It is slightly salty, almost colorless, carrying dissolved ions and small molecules in proportions carefully regulated by the secretory cells of the choroid epithelium. In health, it contains very little protein, nothing like the protein-rich fluid that accumulates in tissues when lymphatic drainage fails. It moves slowly, circulating through the ventricles and around the brain's surface, cushioning the brain against mechanical shock, buffering the chemical environment of the central nervous system, maintaining the pressure balance that keeps the brain floating gently within its fluid-filled space, rather than resting on the hard bone beneath. That floating is something to hold in mind for a moment. The brain, which weighs roughly 3 pounds in air, weighs only a fraction of that in the cerebrospinal fluid that surrounds it, because the buoyancy of the fluid reduces the effective gravitational load on the tissue. The brain sits in its fluid the way a net float sits in water, partially suspended, its true weight mostly borne by the medium around it. Cerebrospinal fluid circulates slowly around it, entering the subarachnoid space from the ventricles, moving over the brain's surface and down along the spinal cord, a calm, continuous motion present every moment of every life, felt by no one, witnessed by almost no one, essential to the functioning of everything inside the skull. What Nedergaard's laboratory showed in the imaging experiments that produced the 2013 paper in the journal Science was that this cerebrospinal fluid was not merely circulating around the brain's surface. It was entering the brain tissue. It was moving inward along the periarterial spaces, the fluid-filled sheaths around the penetrating arteries, and passing through the astrocyte end-foot layer into the interstitial space between neurons and glial cells, where it mixed with the brain's own interstitial fluid and collected the soluble waste that had accumulated there during the brain's operation. And then it moved outward again, draining toward the spaces around the brain's draining veins, converging toward the brain's outer surface, and ultimately leaving the skull through channels that connect to the cervical lymph nodes in the neck, in and through and out, along the arteries inward, through the tissue, along the veins outward. A circuit. The name Nedergaard's group gave to this system placed two words beside each other that had not previously belonged in the same breath: glial, lymphatic. The glymphatic system, a waste clearance network that does what the lymphatic system does for the rest of the body, but built from glial infrastructure rather than dedicated lymphatic vessels, running not through open channels in the tissue, but through the organized architecture of the astrocyte end-foot layer and the periarterial and perivenous spaces it lines. The name holds both halves of what the system is. The glial half names its structure, the lymphatic half names its function, and in the gap between those two words, in the short syllable that has been removed to make the compound work, something important rests: the evidence of a biological solution arrived at by a different route, through the particular constraints of a sealed, barrier-protected, metabolically extraordinary organ, arriving at the same functional endpoint as the lymphatic system through entirely different means. The astrocyte end-foot layer is the key structural element of this solution, not because the end feet form a sealed barrier. They do not seal the periarterial space completely, and the fluid can move through gaps between adjacent end feet as well as through the end feet themselves, but because of what the membranes of those end feet contain. On the surface of the astrocyte end foot, pressed against the outer wall of the blood vessel, there is a protein. Not one copy of the protein, not a handful, but clusters of it, so dense that under electron microscopy the channels appear as a regular lattice, a tightly ordered molecular array covering the vessel-facing surface of the end foot in extraordinary concentration. The protein is a water channel. Its molecular structure is a barrel-shaped pore, just wide enough to allow single water molecules to pass through in a rapid, single-file chain, while blocking ions and most other solutes, selecting for pure water with a precision that no simple membrane pore could achieve. This protein is called aquaporin 4, Water channels are not rare in biology. They are present in the kidneys, where they control the concentration of urine. They are present in the lens of the eye. They are present in red blood cells, in the cells lining the gut, in the cells that form the cerebrospinal fluid itself. Aquaporins are an ancient family of proteins present in organisms from bacteria to plants to mammals, solved once by evolution and then carried forward across hundreds of millions of years because the basic problem they solve, how to move water quickly through a membrane that must remain selectively impermeable, recurs in every watery organism that has ever lived. But in the astrocyte end foot, aquaporin 4 is present at a concentration that has no counterpart elsewhere in the body. The density of the channels on the vessel-facing membrane is far higher than on the other surfaces of the astrocyte. This uneven distribution, high concentration on the side facing the blood vessel, lower concentration on the side facing the brain tissue, is what makes glymphatic transport directional rather than merely diffuse. Water and the solutes it carries are not moving equally in all directions through the end-foot layer. The movement is oriented inward along the arteries, outward along the veins, the gradient maintained by the specific distribution of the channels. The term for this uneven distribution is polarity. Aquaporin 4 polarity, the preferential concentration of the channel at the vessel-facing surface of the astrocyte end foot, is the molecular basis of directed glymphatic flow. Without it, the fluid could move, but it would move without consistent direction, spreading from the periarterial space into the tissue and finding no particular path toward the perivenous drainage routes. With it, the movement of water through the end-foot layer becomes an organized flow, driven by the pressure gradient between the inflow side and the outflow side of the circuit, guided by the specific placement of the channel proteins in the membrane. Think of what this means in terms of the scale at which it operates. The aquaporin 4 channel is a protein, a molecule built from amino acids folded into a specific three-dimensional form, no more than a few nanometers across, perhaps 5 or 10 times the diameter of the water molecules it transports. The astrocyte end foot that holds it is a flat tile of living membrane, a few micrometers wide, larger than the channel by perhaps 1,000 times, but still smaller than the diameter of a human hair. The blood vessel the end foot wraps is perhaps 10 to 50 micrometers across, visible under a standard light microscope as a thin-walled tube, and the brain that all of this serves is the organ sitting just behind the eyes, gray-pink and corrugated, held in the hands with care. From the nanometer to the organ in 4 steps: the channel protein, the end foot, the vessel and its sheath, the whole brain, each scale organized and interlocked, the molecular precision of the smallest level producing the directed bulk flow observed at the scale of tissue and organ. What Nedergaard and her colleagues had found was not just a new physiological system, it was an answer to a question that had not been clearly asked: where does the brain's waste go? And the answer had been there all along, encoded in the distribution of a protein on the surface of cells that wrap every blood vessel in the brain, running every night in every sleeping mammal, clearing the metabolic debt of the day's operation in the dark hours while the rest of the body rests. The experiments that revealed it used 2-photon microscopy, a technique that uses infrared laser pulses to excite fluorescent molecules deep in living tissue without the tissue-damaging energy deposition that shorter wavelengths would produce. The result is a 3-dimensional image of fluorescent tracers moving through intact brain tissue in a living animal, with the kind of spatial resolution that lets the observer watch fluid move along specific anatomical structures in real time. The penetrating arteries appear as tunnels of darker tissue, their walls the boundary between the vessel lumen and the surrounding brain. The periarterial space around them shows up as a bright halo when the tracer arrives, the fluorescence spreading inward from the arterial sheath toward the parenchyma, toward the dense interwoven tissue of neurons and glial cells that makes up most of the brain's volume. That spreading inward was the thing that no one had seen before. Cerebrospinal fluid had been known to exist for centuries. Its circulation around the brain's exterior had been mapped by anatomists long before modern imaging made individual cells visible. But its movement into the brain tissue, the bulk flow from the periarterial space through the astrocyte end-foot layer into the interstitial space, and its continuation through the parenchyma to the perivenous drainage routes on the other side, had not been observed, had not been measured, had not been part of the picture that neuroscience held of how the brain managed its own internal chemistry. The picture that came after looked different. The brain was not sealed from its own cerebrospinal fluid. The fluid entered the tissue, moved through it, and carried something out. The brain was not dependent on the blood-brain barrier alone to manage the soluble debris of its operation. It had a second clearance system, a bulk flow system, organized around the blood vessels and built from the glial infrastructure that was already there for other reasons, or perhaps had always been there, partly for this reason. Partly for this reason. That phrasing is careful because the astrocyte does many things. It regulates the chemical environment of synapses. It provides structural support. It supplies neurons with metabolic substrates. It participates in the formation and function of the blood-brain barrier itself. The aquaporin 4 channels in the end foot are involved in brain-water homeostasis more broadly, in the management of edema after injury, in the regulation of swelling and contraction of the extracellular space. The end foot is not a structure that evolved solely to enable glymphatic clearance. It is a structure with multiple functions, and glymphatic transport is one of them, perhaps an ancient one, woven into the biology of the astrocyte end foot at a time long before the question was asked. The choroid plexus keeps producing its fluid. It does not slow at night. It does not speed up in the day. The secretory cells of the choroid epithelium work continuously, their ion pumps and transport proteins moving water and solutes from the blood into the ventricles at a steady, unhurried pace, maintaining the cerebrospinal fluid volume and composition within narrow bounds across the full span of every hour. 1/2 liter a day in a human, produced and replaced over and over, the old fluid draining away through the arachnoid granulations and the cervical lymphatics, while the new fluid forms behind it. That continuous production is not incidental to the glymphatic system. The choroid plexus is the source of the inflow, the pressure gradient that drives cerebrospinal fluid from the subarachnoid space into the periarterial channels, and from there through the astrocyte end-foot layer into the brain parenchyma, begins with the hydrostatic pressure of the fluid the choroid plexus continuously makes. It is a gentle pressure, nothing like the arterial blood pressure that drives blood through the capillaries. The periarterial space is not a high-pressure conduit. The bulk flow through the parenchyma is not fast, but it is continuous, and over the hours of a night, that continuous low-rate flow carries a significant volume of interstitial fluid through the tissue and outward toward the perivenous draining routes, and everything dissolved in that fluid goes with it. The glymphatic circuit is, in one sense, a very slow river. Its current is measured in microliters per minute, not liters. Its channels are nanometers to micrometers wide, not millimeters. Its driving pressure is the gentle push of continuously produced cerebrospinal fluid, supplemented by the mechanical pulse of arterial walls expanding with each heartbeat, transmitted into the periarterial fluid as a small rhythmic pressure wave. Every beat of the heart produces a small mechanical push into the periarterial space, and that push, repeated 60 to 80 times per minute, night after night, for an entire lifetime, is part of what moves the fluid inward. Slow, small, continuous, mechanical, chemical, a system without sensation, without awareness, without any signal reaching consciousness. The brain does not feel its own drain working. The fluid moving through the periarterial spaces produces no sensation. The bulk flow through the interstitial channels produces no signal. The clearance happens entirely below the threshold of any experience, in the deep interior of the organ that produces all experience, unreported and unnoticed, night after night, for a life below the threshold. That phrase carries something worth holding for a moment. Almost everything the body does is below the threshold of experience. The heart beats 100,000 times a day, and the awareness of it is rarely present. The kidneys filter the blood continuously, and nothing of that filtering reaches consciousness unless something goes wrong. The gut moves its contents through 20 feet of coiled tissue by slow muscular contraction, and the process is as invisible to the mind as the movement of the tides is to the fish in the water below. The glymphatic system is the brain's version of this invisible maintenance. Like every other organ, the brain is not only the seat of the function it is famous for: thought, sensation, memory, language, the whole theater of conscious experience, but also a tissue with a metabolic life, burning energy and generating waste and requiring clearance, like the liver, like the muscle, like the heart. That maintenance requirement does not pause when the brain is thinking. It accumulates with every synapse that fires, with every protein that is synthesized and used and degraded, with every moment of the brain's continuous operation. And at some point, in the hours of each night, when the behavior of the animal that carries the brain permits, the clearance runs. The glial cells are there. They have always been there, wrapped around every vessel, their end feet pressed close, their aquaporin forechannels waiting in the membrane. The cerebrospinal fluid is there. It has always been there, filling the spaces and circulating in its slow continuous round. The periarterial channels are there, not hollow tubes added by evolution for this purpose alone, but the inevitable anatomical consequence of a vessel descending into tissue. The space between the vessel wall and the surrounding tissue, always present, always fluid-filled, waiting to carry whatever the system's pressure gradient would push through it. The imaging experiment that night at Rochester did not create any of this. It revealed it. It placed a tracer in the path of something that had been moving without witness for hundreds of millions of years, and made that movement visible at last, on a screen, in a laboratory, in the slow blue light of the microscope. What the tracer showed was the path, inward along the arteries, through the end-foot layer, through the tissue, outward along the veins, the path that waste takes, the path that cerebrospinal fluid takes when it enters the brain and emerges again, carrying what the brain needed to lose. And the door through which that fluid passes, the membrane at the center of the whole mechanism, the molecular structure that makes directed bulk flow possible without any dedicated vessel infrastructure, is the aquaporin 4 channel, sitting in its dense lattice on the vessel-facing surface of the astrocyte end-foot. It is a small structure, 5 nanometers across, perhaps, a barrel of folded protein its pore too narrow for anything but a single water molecule at a time, its surface amino acids, arranged in a pattern conserved across species so distant from one another that their last common ancestor was 500 million years in the past. The basic form of the aquaporin channel is that old. The brain's use of it, concentrated on the end foot, polarized toward the vessel, organized into the specific distribution that enables directed glymphatic flow, is a more recent arrangement, arrived at through the particular evolutionary pressures of a sealed, barrier-protected, metabolically extreme organ that could not simply borrow the lymphatic solution the rest of the body was using. The rest of the body has its lymphatic vessels, with their smooth muscle walls and their one-way valves and their slow contractions carrying interstitial fluid toward the lymph nodes. The brain has the aquaporin 4 channel, expressed at extraordinary density on a surface that was already there, already organized, already in contact with every vessel in the tissue. The outcome is the same: waste collected from the interstitial space and carried away, achieved through different architecture, scaled to the particular constraints of the structure that needed it. Different route, same ending, the waste moves. The brain stays clean enough to do its work the next day, and the day after that, and for the full span of the life contained within the skull. The choroid plexus is still producing its fluid now, somewhere inside every sleeping brain in every sleeping body. The secretory cells are still at their quiet work, still moving ions and water from the blood into the ventricles, still filling the spaces with the clear, slightly salty fluid that has been serving the brain since long before the brain was capable of knowing that it existed. The fluid circulates. The end feet press against the vessels. The channels sit in the membrane, their pores open, ready for the pressure gradient that will drive water through them and carry the night's clearance forward. The lattice of aquaporin 4 on the surface of the astrocyte end foot is the last thing to hold in mind before the rest of the night arrives. A molecular array, regular and precise, covering the vessel-facing membrane of a cell that wraps every blood vessel in the brain, not a structure a person could ever see. Too small by many orders of magnitude for any sense the body has, but real and present and performing its function right now in the tissue behind the eyes, in the dark, without any witness other than the slow tide of fluid moving through the channels it makes possible. That is the solution the brain arrived at. Not a new vessel system, not a network of tubes, not a parallel circulation requiring its own pumps and its own valves, a protein. A water channel positioned with precision on the surface of a cell that was already there, in the right place, touching the right structure, waiting for the right pressure gradient to begin the work. Old, small, precise, and sufficient. The lattice holds, the fluid moves, the drain runs on. A penetrating artery leaves the brain's surface and begins its descent. It is a narrow vessel, perhaps 1/10 of a millimeter across, carrying oxygenated blood inward from the cortex toward the deep tissue beneath. Around it, pressed close on every side, is brain parenchyma, the dense living mass of neurons and glial cells and their elaborate connections. The artery does not travel through this tissue bare. Around its outer wall runs a sleeve of fluid-filled space, thin as a thread of water caught between two surfaces, following the vessel all the way down from the surface into the deepest structures of the brain. That sleeve is the beginning of everything. It is the entry point of the glymphatic pathway, and the cerebrospinal fluid moving slowly within it will, before the night is through, carry the brain's accumulated waste outward toward places where the body can manage it. These fluid-filled channels surrounding the penetrating arteries have been known to anatomists for more than a century. The Italian anatomist Charles Langer, and later the German pathologist Rudolf Virchow, and the French anatomist Charles Robin each described them in the 19th century, noting their presence around the vessels that dive from the brain's outer surface into deeper tissue. They were called Virchow-Robin spaces, a name that persisted through generations of neuroanatomy without anyone fully understanding what moved through them, or why, or in which direction. They were cataloged and measured and observed, and then, for a long time, not much more was made of them. They were gaps between structures rather than structures in themselves. They were spaces, not channels. Nothing in the anatomy of their time suggested they carried anything on purpose. The vessels they surround do something quiet and powerful with every heartbeat. When the heart contracts and sends a pulse of blood into the arterial tree, each artery expands very slightly outward, a fraction of a millimeter, a brief mechanical bulge traveling down the vessel wall as a pressure wave. In the large arteries of the body, this expansion is visible to the fingertip as the pulse at the wrist. In the penetrating arteries of the brain, the same event happens at a far smaller scale, invisible from outside, imperceptible to anything but the fluid immediately surrounding the vessel wall. That fluid notices. The slight outward expansion of the arterial wall pushes against the cerebrospinal fluid in the periarterial sheath, displacing it inward toward deeper brain tissue with each systolic pulse. The cardiac cycle drives the fluid, not dramatically, not forcefully, but steadily and in one direction, beat after beat, hour after hour, for the whole length of a life. Each pulse adds a tiny increment of inward displacement. The fluid does not rush. It advances by a slow accumulation of small pressures, and it carries with it whatever is dissolved in it, molecules, ions, tracers injected into cerebrospinal fluid for experimental observation, and delivers them progressively deeper into the brain's interior. This is the input side of the glymphatic circuit, and its engine is the heart, operating at a remove, transmitting mechanical energy into the brain's fluid spaces through the walls of the arteries it drives. At the outer wall of the artery, before the fluid can enter the brain tissue proper, it encounters a boundary. That boundary is cellular. It is made of astrocytes, a class of glial cell that gives the glymphatic pathway the first half of its name. Astrocytes are the most numerous non-neuronal cells in the brain, and their functions are various and still not entirely mapped, but one of their structural contributions is an almost uninterrupted cellular sheath around every blood vessel in the central nervous system. Each astrocyte extends a broad, flat process called an end foot that presses flush against the outer wall of the vessel, tiling across the surface in irregular interlocking panels the way stone slabs cover a courtyard, with very little of the vessel wall left exposed. Taken together across every vessel in the brain, these end feet form a structure called the glia limitans perivascularis, the glial boundary around the vessels, a continuous cellular lining that separates the perivascular fluid space from the brain's interstitial space. This boundary is not a wall in the solid sense, it is living membrane, and within that membrane, positioned with a precision that is not accidental, are water channels. They are called aquaporin 4 proteins, and they are present in astrocyte end feet at a density far higher than anywhere else in the brain, far higher, in fact, than in almost any other tissue in the body. They are packed into the end foot membrane so tightly that under electron microscopy they resolve as a regular repeating molecular lattice, a crystalline arrangement of channels pressed side by side. Each one is a narrow protein pore just wide enough for water molecules to pass through in single file, regulated by the physical chemistry of the membrane around it, thousands of them in each end foot, millions across the whole vascular surface of the brain. Their role in the glymphatic circuit is to allow water, and with it the dissolved molecules the water carries, to cross from the perivascular space into the interstitial space of the brain, parenchyma, and back again, far more rapidly than diffusion alone could account for. The channels do not pump, they do not expend energy to move the water. They simply allow it to cross the membrane in response to pressure and osmotic gradients, which the arterial pulse and the drainage system on the outflow side continuously maintain. The astrocyte end foot, with its dense mosaic of aquaporin 4 channels, is the interface between the fluid highway running alongside the artery and the intimate chemical environment of the brain's interior. It is simultaneously the wall and the gate. Once past the end feet, the cerebrospinal fluid that has entered from the periarterial space mixes with the fluid already present between the brain's cells. This interstitial fluid occupies the narrow gaps between neurons, between glial cells, between the densely packed elements of the neuropil, the interwoven mass of axons and dendrites and synaptic terminals that fills the brain's volume. Those gaps are very narrow. Estimates place the width of the extracellular channels between cells at somewhere between 20 and 60 nanometers across, a dimension so small that no light microscope can resolve it, only electron microscopy. In those channels, the fluid is not still. It moves, not by diffusion, which is random and directionless, but in bulk, a slow directed current flowing from the arterial side of the tissue, where pressure is higher, toward the venous side, where it is lower, driven by the same arterial pulse that brought the fluid in from the periarterial sheath. This is the distinction that makes the glymphatic system a clearance system, rather than merely a fluid compartment. Diffusion, left to itself, would be slow and isotropic, molecules spreading outward in all directions from their source, taking hours or days to travel meaningful distances through the dense tissue of the brain. Bulk flow carries everything in one direction, regardless of each molecule's own diffusivity, and does so far faster than passive diffusion could manage. A protein that would take many hours to reach a paravenous drainage route by diffusion alone can arrive there in a fraction of that time when carried by bulk flow. The brain's interstitial fluid is not a pond. Through the night, while the body lies still, it runs like a slow, broad, almost imperceptible river. Moving through the interstitial space, this fluid picks up what the brain's cells have been producing and releasing during the day's activity: metabolic byproducts, fragments of proteins, the molecular aftermath of synaptic signaling. All of it dissolves into the extracellular fluid and waits. The bulk flow of glymphatic transport carries it from the arterial side of the tissue toward the venous side in a slow, continuous sweep, drawing the solute-laden fluid out of the parenchyma and toward the paravenous spaces that form the outflow side of the circuit. On the outflow side, the geometry reverses. The draining veins of the brain are surrounded by their own paravascular spaces, and it is into these paravenous channels that the interstitial fluid, now carrying its load of molecular waste, is collected and channeled. The fluid gathers from across the parenchyma and converges on these venous drainage routes, moving from the deep tissue toward the brain's surface, funneled by the same pressure gradients that drove the inflow. The astrocyte end feet around the veins complete the same functional role they serve on the arterial side, the interface between tissue and channel. But now the direction of net flow is outward, away from the parenchyma, carrying the waste-laden fluid toward the surface of the brain. From the paravenous spaces, The flow continues outward into the subarachnoid space, the fluid-filled layer that cushions the brain's outer surface, lying between the brain and the arachnoid membrane that lines the inside of the skull. The subarachnoid space is bathed in cerebrospinal fluid, and it is a place of slow, continuous movement. Fluid enters it from the ventricles within the brain, circulates across the brain's outer surface, and drains away through various exit routes that carry it toward the body's peripheral circulation. It is a wide, calm space relative to the narrow channels of the parenchyma, and the fluid arriving there from the perivenous drainage routes enters a broader current. The subarachnoid space is not the end of the circuit's outward journey. The fluid that has carried the brain's waste from the depths of the neuropill out to the surface must leave the skull entirely to complete its role. Several drainage routes accomplish this. The most studied in recent years runs through the cribriform plate, the thin perforated bone that forms the roof of the nasal cavity, where nerve fibers from the olfactory system pass through to the mucosa below. Alongside those nerve fibers, cerebrospinal fluid can cross from the subarachnoid space into the nasal submucosa, and from there into the cervical lymphatic vessels. Other routes run along the roots of cranial nerves and along the spinal cord, where lymphatic connections along the nerve sheaths provide additional drainage pathways. The drainage is not through a single exit, but through several, distributed around the base of the skull and the upper spine. At the end of these routes, outside the skull, lie structures that belong entirely to the peripheral immune system. The cervical lymph nodes, small oval nodules of lymphoid tissue, distributed along both sides of the neck, felt sometimes during illness as tender swellings, receive this drainage. They are not specialized brain structures. They are the same lymph nodes that drain fluid from the face, the scalp, the throat. The brain's glymphatic outflow arrives among these familiar peripheral immune tissues without ceremony, and there it encounters immune cells that can inspect the molecular cargo it carries, clear pathogens if any are present, and process the molecular debris the brain has expelled during the night. The circuit that began in the periarterial spaces deep inside the brain closes far outside the skull in a small knot of tissue below the jaw. The structural logic of this circuit becomes clearer when considered against the constraints that shaped it. The brain sits inside the skull, a rigid enclosure of fixed volume. Unlike every other organ in the body, it cannot swell to accommodate the kind of lymphatic vessel infrastructure that drains other tissues. Conventional lymphatic capillaries, the vessels that collect interstitial fluid from muscle and connective tissue and carry it toward lymph nodes, require physical space to run alongside blood vessels. They exist in essentially every tissue in the body, but not in the brain itself. The brain's own tissue contains no lymphatic vessels, and the blood-brain barrier, the tight junctions between endothelial cells lining the brain's capillaries, prevents the kind of molecular traffic that peripheral lymphatic vessels are designed to handle. The brain is, in the anatomical sense, sealed against the immune surveillance machinery that monitors the rest of the body. The glymphatic system is the solution the brain arrived at, using the vascular architecture already present, routing cerebrospinal fluid along the outside of the arteries and veins that penetrate the brain's tissue, and deploying a specialized class of glial cells, whose end feet densely line every vessel wall. The brain constructed a clearance pathway from structures it already had. The arteries provide the pumping force, the periarterial channels provide the inflow route, the astrocyte end feet and their aquaporin 4 channels provide the crossing point into and out of the interstitium, the paravenous channels provide the outflow route, and the cervical lymph nodes, outside the skull provide the peripheral immune connection the brain would otherwise lack. What makes the system precisely functional rather than merely structurally plausible is a property called polarity. The aquaporin 4 channels in astrocyte end feet are not distributed uniformly across the whole astrocyte surface. They are concentrated specifically on the face of the end foot that presses against the vessel wall. This preferential positioning, far more channels on the perivascular face than on any other membrane surface of the astrocyte, is what gives glymphatic flow its directionality through the cell layer. Water and solutes are conducted across the end foot membrane, specifically at the point of contact with the perivascular space, so that fluid moves in and out of the interstitium at the vascular interface, rather than leaking randomly across the whole cell surface. The polarized distribution of the channels is not a structural accident. It is the feature that makes the system work as a directed circuit rather than a generalized leak. This polarity is maintained by molecular scaffolding proteins inside the astrocyte, particularly a protein called alpha-syntrophin, which anchors aquaporin 4 channels to the perivascular membrane face and holds them in position. When this scaffolding is disrupted, the channels redistribute. They spread more evenly across the astrocyte surface. Their concentration at the vessel face falls, and the directional advantage that makes glymphatic clearance efficient is reduced. The fluid can still cross the cell membrane, but the precise routing that distinguishes bulk flow clearance from simple diffusion is degraded. The system becomes less a directed channel and more a slow seepage. Disruption of aquaporin 4 polarity happens in known biological contexts, and the contexts matter. Normal aging is one of them. As the brain ages through the decades of middle and later life, the anchoring proteins that hold aquaporin 4 channels in place at the perivascular membrane face become less effective. The channels spread. The polarized architecture of the astrocyte end foot, so precisely maintained in the young adult brain, gradually loses its organization. This is not a pathological process in the sense of being caused by disease. It is part of the brain's ordinary structural aging, proceeding slowly in every brain that lives long enough, but the functional consequence is a progressive reduction in glymphatic clearance efficiency, emerging over years, measurable in animal models as a declining rate of cerebrospinal fluid influx into the parenchyma. Brain injury is another context in which polarity is disrupted, and more acutely. Following a traumatic injury, astrocytes respond with a rapid structural change. They swell, their processes thicken, the organized architecture of the end feet at the vessel wall is compressed and distorted. The aquaporin 4 channels that should be held in tight array at the perivascular membrane face are displaced by the swelling and the structural reorganization that follows injury. In the acute phase after trauma, glymphatic function in the injured region falls sharply at a time when metabolic demand is high and inflammatory proteins are accumulating in the extracellular space. The very pathway that would normally clear them is the most compromised. The relationship between polarity and function makes the glymphatic system more structurally delicate than its basic anatomy suggests. A pathway that routes fluid along arterial sheaths, through astrocyte end feet, across the interstitium, and out along venous channels, seems robust, a physical system driven by hydraulic pressure. But the efficiency of that system depends at every crossing point on the precise molecular organization of the membrane through which the fluid passes. The position of the water channels within the end foot membrane is as essential to glymphatic function as the channel's existence. Architecture matters at the molecular scale. The difference between a working drain and an inefficient one may be the difference between channels held tightly in position by their scaffolding and channels that have drifted a little from where they need to be. The interstitial space itself, the narrow corridors between cells through which the bulk flow runs, has its own structural character. Occupying roughly 15 to 20% of total brain volume in the spaces between neurons and glia, it is not empty. It is filled with a loose matrix of large structural molecules, proteoglycans, hyaluronic acid, and other extracellular matrix components that form a physical scaffolding through which the fluid must percolate. The geometry of this matrix, its mesh size and charge properties, influences how easily solutes of different sizes and charges can move through it. Large aggregating proteins move differently than small, highly soluble metabolites. The extracellular matrix is not a passive substrate, but an active participant in determining what the bulk flow can carry and how efficiently it can do so. The pressure gradient that drives flow from the arterial side of the parenchyma to the venous side is modest by the standards of most hydraulic systems. It is generated by the combination of arterial pulsation pushing fluid inward on the inflow side and the lower pressure of the paravenous drainage channels pulling fluid outward on the exit side. These pressures are small, a fraction of the systemic blood pressure, and the resistance they must overcome in the narrow extracellular channels is correspondingly significant. The bulk flow that results is slow by any ordinary measure. Tracers injected into cerebrospinal fluid require time, measured in minutes to hours, depending on the depth of the tissue, to travel from the periarterial entry point to the perivenous exit. This is much faster than diffusion alone would accomplish, but it is not a rapid flush. It is a slow, continuous sweep that runs without interruption across the full span of every day and night, and whose total volume moved over hours is substantial, even though any single molecule's transit is unhurried. The circuit runs in a loop that, if traced completely, crosses 3 different fluid compartments. It begins in the cerebrospinal fluid of the periarterial sheath. It crosses into the interstitial fluid of the brain parenchyma through the astrocyte end feet. It exits into the cerebrospinal fluid of the perivenous spaces and the subarachnoid space. And then it drains to the lymphatic vessels and lymph nodes of the neck outside the skull. No single compartment is isolated from the others. The system that maintains brain function is also the system that connects the brain's internal chemistry to the peripheral immune system through a circuit of quiet hydraulics. The fluid doing all of this work is produced far from where it flows. Deep within the brain's interior, folded inside the 4 hollow chambers called ventricles, lies a tissue whose sole function is continuous fluid secretion. The choroid plexus, named for the blood vessel-rich folds that give it the appearance of a dense tangle of vascular branches, sits in each ventricle, pressed against the ventricular wall, and produces cerebrospinal fluid from blood plasma by a combination of filtration and active transport. The process runs continuously. The choroid plexus secretes roughly half a liter of cerebrospinal fluid over the course of every day. In humans, a substantial volume, given that the total cerebrospinal fluid volume in the brain and spinal cord at any moment is only around 100 to 150 milliliters. The entire cerebrospinal fluid volume is replaced several times each day, a rate of turnover that ensures the fluid passing through the glymphatic pathway is consistently fresh. From the ventricles, cerebrospinal fluid flows outward through narrow passages into the subarachnoid space, where it bathes the brain's outer surface before draining through the various exit routes already described. Some of it enters the periarterial spaces from the subarachnoid space, following the penetrating arteries inward as they descend from the cortex into deeper structures. The driving force for this inward entry is partly the arterial pulsation already described, and partly a slower, broader pressure differential between the subarachnoid space and the parenchymal interstitium. The fluid does not simply sit in the subarachnoid space. It is continuously pulled inward along the arterial sheaths and pushed outward again through the venous drainage routes, cycling through the tissue in a slow but unceasing circuit. The arteries that the fluid follows inward are not uniform in size. The largest penetrating arteries descend from the brain's surface into the subcortical white matter and deeper gray matter structures, the basal ganglia, the thalamus, the brainstem nuclei. Smaller arterioles branch from these parent vessels as they descend, reaching the finest capillary beds embedded within the parenchyma. The periarterial spaces follow the arteries and arterioles inward, growing narrower as the vessels narrow, until at the capillary level, the formal perivascular space is no longer anatomically distinct, and the fluid transport transitions more fully into the interstitial bulk flow. The circuit therefore covers the brain from its surface to its deepest structures, reaching tissue that no surface drainage mechanism could serve. Along the routes from the deep parenchyma back toward the surface, the perivum spaces follow the veins that collect deoxygenated blood from the tissue. The veins course from the deep white matter and subcortical structures toward the cortex, and from there to the brain's surface, and the perivenous drainage channels run alongside them. The fluid that has collected metabolic waste from deep in the neuropill travels back toward the surface in these channels, merging progressively into larger drainage routes as the veins converge. From the cortical surface, it enters the subarachnoid space, and from there, the various outflow pathways to the lymphatics below the skull. The cervical lymph nodes that receive this outflow are small and sit quietly in the soft tissue of the neck, unremarkable in size and appearance, indistinguishable by surface anatomy from any other lymph node in the body. The fluid arriving in them carries what was dissolved in the brain's extracellular space: proteins, lipids, fragments of cellular metabolism, and whatever other soluble material the bulk flow picked up during its passage through the tissue. The lymph node's resident immune cells begin to process this cargo, sampling what the brain has sent out. The brain is an immunologically privileged tissue, sheltered behind the blood-brain barrier, and largely separated from peripheral immune surveillance. But through this drainage route, slow and indirect, traveling hours from deep cortical tissue to a cervical lymph node, some molecular information about the brain's internal state reaches the peripheral immune system each day. This is not immune surveillance in the rapid and responsive sense of the peripheral immune system's usual operation. It is something slower, more like a deferred report than a live signal. The brain does not broadcast its internal chemistry to the body with any urgency. The glymphatic drain carries its load outward in the quiet hours, and the lymph nodes receive it in their own time. There is no immune activation, no inflammatory signal under ordinary conditions, just the routine processing of familiar molecular debris, the metabolic residue of a functioning brain, handled and dispersed. The circuit completes, not with drama, but with the ordinary work of tissue maintenance at the end of a long and specific journey from deep inside the skull to a small and unremarkable nodule in the neck. To trace the full structural path from beginning to end is to follow something that travels a long way in a confined space. Starting within the periarterial sheath of a penetrating cortical artery, a channel perhaps 20 to 30 micrometers wide, surrounding a vessel descending into the parenchyma, The cerebrospinal fluid carries its inward journey downward through the cortex, past the white matter, towards subcortical structures. Crossing through the astrocyte end-foot layer, it enters the interstitial space, where its transit slows to the pace of extracellular bulk flow, moving through channels a fraction of a millimeter wide across a tissue that may be several centimeters deep. Collecting waste along the way, it exits through paravenous channels, rises back toward the cortical surface, enters the subarachnoid space, and then crosses the skull base through the cribriform plate or along nerve sheaths, descending through the tissue of the nasopharynx and the upper neck to arrive, eventually, in a lymph node resting against the carotid sheath below the jaw. The total distance from entry to exit from the periarterial sheath at the cortical surface to the cervical lymph node below the skull is perhaps 15 centimeters in a human brain. For a fluid moving in bulk, flow through spaces measured in nanometers to micrometers, this is an enormous structural journey. The time required in a healthy sleeping brain may be several hours. The circuit does not announce itself. It produces no sensation. It leaves no trace in ordinary experience. It has been running without notice since the earliest weeks of fetal brain development, and it will continue running at rates that shift slowly with age and circumstance until the end of the brain's functional life. What holds the whole system together, what ensures that the fluid follows the circuit from entry to exit rather than dispersing randomly through the tissue, is the structural geometry of the pathway combined with the molecular precision of the astrocyte end feet. The periarterial and perivenous channels are low-resistance routes along which fluid can travel more easily than across the high-resistance interstitium. The pressure gradients favor movement along these channels rather than perpendicular to them. The aquaporin 4 channels in the end feet concentrate the crossing points at the vascular interface, ensuring that the fluid's entry into and exit from the interstitium is mediated precisely. The extracellular matrix provides the structural frame through which the interstitial bulk flow runs. Each component of the architecture contributes to the directional integrity of the circuit. The glymphatic pathway runs through a brain that is from the outside entirely still. In sleep, the body does not move. The skull is a sealed and rigid space, dark and quiet. Within it, the fluid circuits run their slow directional rounds, the arterial pulse transmitting mechanical energy into the periarterial fluid, the cerebrospinal fluid advancing inward along the penetrating vessels, the astrocyte end feet passing it through at the vascular surface, the interstitial bulk flow carrying it across the parenchyma in a slow and continuous current, the paravenous channels collecting it and routing it back toward the surface, the subarachnoid space gathering it before it drains away through the base of the skull. All of it proceeding in a structure that has been present in the brain since before birth, operating through mechanisms that had not been imaged or named until recently, doing a work that is as old as vertebrate nervous systems and as quiet as anything the body does. The cervical lymph nodes, receiving the outflow of this long circuit, rest in the neck below the jaw. They are small. They lie still in the soft tissue, receiving what the brain has sent down to them through the hours of the night, filtering the fluid that entered the brain as clean cerebrospinal fluid and exits it carrying the molecular residue of the day's neural activity. The circuit that began in the deep channels of the brain's periarterial spaces closes here, in a small and unremarkable node of lymphoid tissue, at the end of a structural journey that begins inside the skull and ends beyond it, the brain's own pathway to the drain, running quietly through the dark. On the recording, the wave comes first as a broad, slow rise, a swell lifting from the baseline, climbing high above it, then falling again, then rising once more, each cycle completing itself in roughly a third of a second, unhurried, repeated, the whole trace moving from left to right across the paper in a long rolling rhythm that looks less like the output of an active brain and more like the surface of a calm sea in a distant swell. This is the electroencephalogram of a sleeping person deep in their night. These are delta waves. Their frequency sits below 4 cycles per second, usually closer to 1 or 2, and their amplitude, the height of each wave above the quiet baseline, is greater than in any other stage of sleep, greater than in wakefulness. The tallest, most synchronized electrical signature the brain produces across the whole span of a life. The brain generating these waves is not doing nothing. That much is certain now, though for most of the history of sleep science, the opposite assumption held, that the sleeping brain was simply a waking brain with the lights turned down. What the delta wave actually marks is a state of extraordinary internal coordination, millions of neurons across wide territories of cortex firing and falling silent in slow, sweeping unison, a biological rhythm as regular and as deep as breathing. Something essential is happening in the tissue that produces this wave, something the waking brain cannot do or will not do, or something the waking brain is simply too busy to begin. The cleaning starts here. In the slow, high waves of deep sleep, in the quiet that the recording makes visible, a process opens that has been waiting since the morning. Before that process can be understood, there is a geometry to consider. The brain sits inside a rigid case of bone, fixed in volume, unable to expand. Every structure inside it, every neuron, every supporting cell, every blood vessel, every channel of fluid must coexist within that fixed space, and any change in one structure comes at the expense of something else. During wakefulness, the neurons and their supporting cells are plump and close-packed, their outer membranes pressed near to one another, the narrow corridors between them reduced to channels perhaps 20 to 60 nanometers wide. A nanometer is a billionth of a meter. A red blood cell is roughly 7,000 nanometers across. The gaps between brain cells during waking hours are narrower than the wavelength of visible light, so close that the space between them is less a corridor than a crack. Through those cracks, fluid moves. It moves slowly, by diffusion, molecules spreading outward from wherever they are most concentrated toward wherever they are less so. The same process that carries heat from a warm hand into cool air, patient, undirected, governed by nothing more than the mathematics of probability. During waking, this diffusion is the primary mechanism by which the brain's extracellular fluid shifts, and diffusion across the distances that matter in the brain is not fast enough. Metabolic waste produced deep in the tissue, the byproduct of every synapse that fires, every protein that is built and then broken, every hour of sustained thought or sensation, accumulates faster than diffusion can carry it away. The brain runs a deficit during the hours it is used, and that deficit must be repaid. What repays it is not diffusion at all. What repays it is something more like a tide. The tide begins when slow-wave sleep deepens. Across the brain, something changes in the cells themselves. The neurons and their supporting cells, the astrocytes in particular, those branching glial cells that wrap every blood vessel in the brain in a tight cellular sleeve, begin to contract. Not dramatically, not all at once, but measurably, their membranes drawing inward, their volume decreasing, the physical space they occupy within the skull becoming incrementally slightly less. And because the skull does not change volume, what those cells vacate must be filled by something else. What fills it is fluid, The extracellular space, the network of narrow corridors between cells, widens. During wakefulness, it occupies roughly 14% of the brain's total volume. During slow-wave sleep, it expands to nearly 23%. 9 percentage points of additional volume, distributed through every microscopic gap between every cell in the brain. The corridors widen from hairline cracks into channels that, while still invisible to any ordinary sense, offer dramatically less resistance to fluid moving through them. Bulk flow becomes possible. The slow tide can begin. The numbers carry weight here. A shift from 14% to 23% is not a subtle change in a minor parameter. It is a 60% expansion of the space available for fluid movement, occurring across the entire brain within the first hour of deep sleep, every night, reversing entirely upon waking. The sleeping brain and the waking brain are not the same brain in different states of electrical activity. They are, in a physical and measurable sense, differently shaped. What drives that shaping is not fully settled. The best current picture involves ion channels and transporter proteins in the membranes of glial cells, molecular machinery that moves charged particles across the cell wall, drawing water with them through osmosis, the same force that makes a grape swell in water or a raisin shrink in dry air. When glial cells export ions during sleep, water follows, the cells shrink, and the space between them opens. The precise signals that trigger this export at the moment of sleep onset are still being worked out, and what is known sits close beside what is still only suspected, without a clean line between them. What is clear is the outcome. The corridors widen, and the fluid moves. The fluid moving through those widened corridors enters from the brain's surface, driven inward along the sheath surrounding the penetrating arteries, the blood vessels that descend from the cortex toward the brain's deeper structures, each one wrapped in a fluid-filled channel, a sleeve of cerebrospinal fluid, pressed between the vessel wall and the surrounding brain tissue. Arterial walls pulse with each heartbeat, expanding slightly outward as a pressure wave travels through them, and that expansion pushes the fluid in the sleeve forward, inward, toward the brain's interior, one small pulse per heartbeat, perhaps 60 or 70 times each minute through the night. Each individual pulse is tiny. Across hours of sleep, across a full night of slow-wave cycling, the cumulative effect is not tiny at all. The fluid driven inward is cerebrospinal fluid, clear and almost saltwater, simple in its composition, produced in the brain's deep ventricles and circulated through the spaces surrounding the brain and spinal cord. As it enters the tissue along the periarterial sleeves, it encounters the astrocyte end feet, those flat tile-like cellular structures that form an unbroken sheath around every brain blood vessel, pressed close against the outer wall, their surfaces densely packed with protein channels built specifically for water. Through those channels, the cerebrospinal fluid crosses from the periarterial space into the broader interstitial space of the brain, mixing with the fluid that has been there all day, beginning to move through the widened extracellular corridors in bulk, from the arterial side of the tissue toward the venous side, from inflow to outflow, carrying dissolved material along with it in a slow directional current. This is the tide, not diffusion, not random molecular wandering, but directed bulk flow, fluid moving through the tissue in one general direction, sweeping the interstitial space the way a river sweeps its bed, carrying whatever is dissolved in it toward the drain. During slow-wave sleep, in rodents, the rate of that sweeping is roughly 10 times what it is during wakefulness. Not incrementally greater, not modestly improved, 10 times. The brain in deep sleep is performing waste clearance at a rate that the waking brain cannot approach, a difference so large that the waking brain's clearance capacity, measured against it, appears almost vestigial, a maintenance trickle, sufficient perhaps for brief resting periods, but wholly inadequate to the full scale of daily metabolic debt. In rodent studies, when cerebrospinal fluid tracers are injected and their movement through the brain is tracked, the contrast between the sleeping state and the waking state is visible as a difference in how far and how fast the tracer spreads. During wakefulness, the tracer advances slowly, spreading outward from its injection point in an irregular diffuse pattern, unable to penetrate deeply into the parenchyma, remaining largely confined to the periarterial channels near the surface. During sleep, the same tracer moves inward rapidly, spreading through the tissue in a broad coordinated front, reaching deep structures, covering territory the waking state tracer never touched. The image is unambiguous. The sleeping brain is doing something the waking brain is not doing. What determines this tenfold difference is not a single switch, but a convergence of factors, each one necessary, none of them sufficient alone. The expansion of the interstitial space reduces flow resistance and allows bulk movement that diffusion could never accomplish. The continued arterial pulsation provides the mechanical pressure that drives fluid inward. And something else, something chemical, something that threads through the whole sleeping brain like a tide receding from a beach, plays a role that no anatomical change fully explains on its own. In the brainstem, roughly at the level where the brain stalk narrows before entering the spinal cord, there is a small, dense cluster of neurons, compact, closely packed, their cell bodies darkened by the presence of neuromelanin, a pigment that gives the cluster its characteristic, deep color, visible even to the eye when brain tissue is sectioned. This cluster has a name, the locus coeruleus. It is, in a functional sense, one of the brain's master levers of arousal. Its neurons release norepinephrine, a chemical messenger that reaches, through branching axonal projections, almost every other region of the brain, tuning the entire organ toward alertness, readiness, response. When the locus coeruleus fires vigorously, norepinephrine washes across the cortex and the subcortical structures alike. The tone of the entire brain rises, attention sharpens, and every neural system orients outward toward the world. During wakefulness, the locus coeruleus fires steadily, maintaining that chemical tone. During the descent into deep sleep, something quiets it. The firing rate slows, then slows further, the chemical tide of norepinephrine receding progressively, until in the deepest phase of slow-wave sleep, the locus coeruleus is nearly silent, its neurons at their lowest rate of discharge, the norepinephrine signal at its nightly minimum across every territory it reaches. The receding norepinephrine appears to be one of the key molecular signals that opens glymphatic flow. The precise mechanism is not yet fully resolved, but the available evidence points in a consistent direction. Norepinephrine, in addition to its well-known arousal functions, appears to actively suppress the glymphatic pathway during wakefulness, likely by keeping cells swollen and interstitial channels narrow, maintaining the brain in a configuration optimized for neural signaling rather than fluid clearance. When norepinephrine withdraws during deep sleep, that suppression lifts, cells contract, spaces widen, and the glymphatic tide rises. This is a useful way to hold the relationship between wakefulness and sleep. Not that sleep adds something the waking brain lacks, but that sleep removes a suppression the waking brain maintains. The glymphatic system does not wait to be switched on, it waits to be released. And it is norepinephrine, the same molecule that keeps the brain alert and responsive across the waking day, that holds the gate closed until sleep arrives. Something similar, viewed from a different angle, is visible in the behavior of the cerebrospinal fluid itself during the sleep cycle. Not only does flow increase during slow-wave sleep, but the rhythm of that flow couples with the slow oscillations of the delta wave itself, the electrical pulse of the sleeping brain appearing to coordinate the movement of fluid through it. The neural rhythm and the hydraulic rhythm running together in a synchronized pattern. This coupling, still being characterized, suggests that the glymphatic system is not merely a passive beneficiary of the brain's sleeping state, but may be actively organized by the same neural machinery that generates the slow wave. The brain, in some functional sense, may be orchestrating its own cleaning schedule from within. The slow oscillation of the delta wave runs at roughly 1 cycle per second. In deep sleep, 1 rise and 1 fall per second. The whole cortex sweeping through the cycle together, synchronized across territories that during wakefulness are independent in their activity. Each cycle has a brief active phase, during which neurons fire, and a brief quiet phase, during which they fall nearly silent. The pressure in the interstitial fluid may fluctuate slightly, with each cycle rising during the active phase, falling during the quiet, and that fluctuation may contribute to the bulk flow that sweeps waste through the tissue. The brain breathes, in a sense, with its own slow electrical respiration, and the fluid moves with each breath. The relationship between sleep stage and glymphatic activity is not uniform across the whole night. Slow wave sleep, the deep delta wave stage, is when glymphatic clearance peaks, and slow wave sleep is most abundant in the first half of the night, before the sleep cycle tilts progressively toward lighter stages and toward the state that occupies a different and stranger territory altogether in the second half of the night, when brain activity takes a different character, more active, the eyes moving beneath the lids, the body held still by a separate neural mechanism, while the mind produces images and sequences of experience. The glymphatic system does not appear to achieve the same magnitude of clearance. The brain is metabolically busy in that state, its activity patterns in some ways resembling wakefulness, and the norepinephrine fluctuations in that stage are different in character from the sustained withdrawal that accompanies deep slow-wave sleep. The current evidence suggests that different sleep stages serve different restorative purposes, and that the deep slow-wave phase carries a particular responsibility for glymphatic waste clearance that the other phases do not equally share. This distinction matters because sleep is not uniform, and not all hours of sleep are equivalent in their restorative function. A night in which slow-wave sleep is repeatedly interrupted by noise, by light, by temperature, by anything that fragments the depth and continuity of the early night stages, is a night in which the glymphatic window is shortened, its peak activity truncated, its clearance incomplete. The brain wakes the next morning having swept less than it needed to sweep, carrying forward a slightly larger burden than it would otherwise carry, and the chemistry of the interstitial space begins the next waking day from a position that is not fully restored. In animal studies, this accumulated burden is directly measurable. When mice are kept awake, gently prevented from sleeping by environmental novelty and mild stimulation, the concentration of amyloid-beta protein in the brain's interstitial fluid rises steadily throughout the period of wakefulness, accumulating in the extracellular space faster than any waking state clearance mechanism can remove it. After a night of undisturbed sleep, those concentrations fall. After a night of interrupted or shortened sleep, they fall less or not at all, leaving behind a residue above the normal resting level. The brain's accounting system is precise. What is not cleared during the first night does not vanish. It is carried forward into the next waking day, adding to what is produced during that day, compounding toward a level that sustained across many nights and many months begins to approach something more serious than a single sleepless night. In humans, cerebrospinal fluid drawn from people after a night of total sleep deprivation contains measurably elevated concentrations of amyloid-beta compared to the same fluid drawn after normal sleep. Brain imaging using positron emission tomography, a technique that can detect the accumulation of amyloid deposits in living tissue, shows increased amyloid signal in the frontal and parietal regions after sleep deprivation, regions that are also among the first to show amyloid pathology in the early stages of certain neurodegenerative processes. The human data and the animal data point in the same direction, across different methods, across different species, and the picture they assemble is consistent. Sleep deprivation does not merely leave the brain tired, it leaves the brain dirtier. It leaves the interstitial space carrying a heavier load of molecular waste than a properly sleeping brain would carry, a difference that is measurable in a test tube and visible in a scanner, a difference that is real and physical and accumulates over time. What happens to the brain's glymphatic infrastructure when sleep is chronically shortened is more troubling still and slower to resolve than a single night's chemistry. The channels through which glymphatic fluid moves, the perivascular spaces, the astrocyte end-foot layer, the aquaporin 4 proteins through which water crosses into the interstitial space, are not static structures that simply open and close on demand. They are living tissue, and living tissue responds to how it is used. Aquaporin 4, the water channel protein that studded so densely on the vessel-facing surface of every astrocyte end-foot, is not simply present or absent. Its position matters. Under normal conditions, the channels are concentrated on the side of the astrocyte end-foot that faces the blood vessel, specifically oriented to permit efficient directional water transport from the perivascular space into the interstitial tissue and back again. This orientation is called polarity, polarized. AQP4 channels in the correct position make glymphatic flow efficient. Depolarized channels, scattered across the astrocyte's membrane without the precise orientation that concentrates them at the vessel interface, still allow some water transport, but the directional efficiency of the system is impaired. The tide becomes less a sweep and more a diffuse seepage. In animal models of chronic sleep restriction, AQP4 polarity is measurably disrupted. The channels drift from their precise position at the vessel-facing surface, redistributing across a wider area of the astrocyte membrane, losing the tight concentration at the perivascular interface that makes bulk flow directional and efficient. The same disruption appears in older animals and in animals following brain injury, and the pattern is consistent enough across these different conditions to suggest that AQP4 polarity is vulnerable, that it is a precisely arranged structure that can be disarranged, and that its disarrangement has functional consequences for the system it enables. What makes this sobering is not the single night of lost sleep. What makes it sobering is the word that follows chronic. In animals chronically restricted to a fraction of their normal sleep, studies in which this restriction runs across days or weeks rather than a single experimental session, AQP4 polarity disruption does not fully resolve when normal sleep is restored. The architecture of the glymphatic system appears to retain something of the damage, to carry forward a structural alteration that recovery sleep does not simply undo, the way a single night of good sleep undoes the chemistry of a single sleepless night. The perivascular channels themselves may also be altered by chronic sleep loss, their geometry subtly changed by repeated cycles of the edema and inflammatory signaling that accompany sleep-deprived brain states. The precise mechanisms are still being worked out. What is known is the direction. Chronic sleep restriction does not merely reduce how much clearance happens on any given night. It may reduce the brain's capacity for clearance on all subsequent nights, eroding the infrastructure through which the cleaning is done. This is the kind of finding that the style of its measurement barely conveys. In a paper, it is a bar graph showing a statistical difference in immunofluorescence signal between two groups of mice. In a life, it is the slow, invisible accumulation of what was never cleaned away. Not in any single dramatic night, but across the years of a sleep pattern that was never quite sufficient, that took the first hour of slow-wave sleep and cut it short repeatedly, that allowed the body to rest but not to clean with the full efficiency it was built for. The brain built to last decades, perhaps a century, designed by long biological time to be cleared nightly of its own metabolic debris, will perform that task with less thoroughness across every night it is not permitted to clean properly. What accumulates in the interstitial space across those nights does not announce itself. It has no single symptom, no one morning when the shortfall is revealed. It gathers quietly in the extracellular corridors between cells, molecule by patient molecule, in a silence the waking brain cannot hear and cannot feel. The delta wave rolls across the recording, 1 cycle per second, unhurried, the amplitude high and regular and slow. The locus coeruleus rests near silence, its norepinephrine signal at its lowest ebb, the tension it normally maintains across the waking brain released, the glymphatic channels open, the fluid moving through the widened corridors between cells in the slow directional current that deep sleep alone permits. The astrocyte end feet, their aquaporin 4 channels aligned precisely at the vessel interface, pass the fluid through in one direction, inward along arterial channels, outward along venous ones, the whole hydraulic system running at its peak efficiency, the sweeping done, the night's account being settled. This is not metaphor. The current is real, the fluid moves, the waste is carried outward, and the brain that wakes after a full, undisturbed night of deep slow-wave sleep wakes into a slightly cleaner version of itself. The interstitial space, a little more open, the molecular burden, a little lighter, the chemistry of the extracellular environment restored closer to the baseline from which efficient function begins again. There is nothing dramatic in this, nothing visible. The person sleeping does not feel the fluid moving, or the cells contracting, or the channels opening as norepinephrine withdraws. There is only the slow wave on the recording, the quiet of the room, the steady rhythm of breath, and beneath all of it, invisible, patient, working with a precision that required billions of years of biological time to arrive at nightly cleaning that the brain cannot perform in any other state. The astrocyte end feet hold their position at the vessel wall, their channels dense and properly oriented, their polarity intact. Through each one, water passes in both directions, but on net, during deep sleep, the flow is directional. The tide comes in along the arteries. The tide goes out along the veins. In between, the interstitial space of the brain, the narrow, dark corridors between neurons and between glial cells, where the chemical residue of a day's thinking accumulates, is swept slowly, completely, by a current of clear fluid that has been making this journey every night since before the brain. It cleans had language to describe it. The delta wave rises, it falls, The current continues. The night holds still around it. What remains when this infrastructure is slowly worn down, when the channels are not quite as open as they should be, when the astrocyte channels are not quite in the position where they work best, when years of insufficient slow-wave sleep have left a structural alteration that does not fully resolve, is not something felt in a single morning. It is a narrowing, a slight reduction in the thoroughness of each night's cleaning, a slow accumulation of what should have been carried away. The corridors are not closed. The fluid still moves. The system still functions. But the difference between a system functioning at full capacity and a system functioning at reduced capacity is not felt in the moment. It is measured across time in the chemical environment of the interstitial space, in the concentrations of what was left behind, molecule by molecule, night by night, in the dark and silent spaces between cells, where the brain quietly carries what it could not clean away. The delta wave slows as the first cycle of deep sleep nears its end. The locus coeruleus has been quiet for over an hour, its neurons resting, the norepinephrine signal low, and even across the whole brain. In a few minutes, the sleep cycle will shift, and the slow wave will give way to something lighter, and the glymphatic tide will ebb, and the corridors between cells will narrow slightly, as the cells themselves expand back toward their waking volume. For now, the deep wave continues. The fluid moves in its slow, directed current through the widened extracellular space, carrying the night's accumulation outward toward the paravenous channels, toward the subarachnoid space, toward the lymph nodes in the neck, where the drain eventually exits the skull entirely. The brain rests. The cleaning continues. The night holds the whole long process in silence, as it has done every night since the first of these brains was built, and as it does even now, in the quiet of this moment, behind closed eyes. In the narrow space between two neurons, in the dark interior of a living brain, a small molecule drifts. It is not large, it is not unusual. It is a peptide, a short chain of amino acids, produced as an ordinary consequence of the brain doing what brains do, firing, signaling, maintaining the steady electrical commerce of thought and sensation and the quiet background hum of being alive. The molecule is called amyloid-beta, and in a healthy brain, at this moment, it is simply part of the inventory, not a threat, not an anomaly, a byproduct, the way heat is a byproduct of an engine running well, present in the interstitial fluid of every healthy brain at every waking hour, drifting slowly in the narrow corridors between cells, waiting to be carried away. It is being produced right now, in every brain that is awake, in every brain that is working, which is every brain, without pause. Amyloid-beta is being released into the extracellular space as a natural downstream consequence of synaptic activity. The synapses fire. The transmembrane proteins process their signals, and as part of that processing, small fragments are cleaved away, released into the fluid outside the cell, and added to the slow accumulation that the interstitial space holds at any given moment. The production never stops. What varies is the clearance. What changes fundamentally and consequentially is how efficiently the fluid around those cells moves and carries and clears. That clearance is the subject of what follows, not the failure of it, not yet, but the thing itself, the molecular inventory that the sleeping brain methodically removes each night, the specific substances that bulk glymphatic flow collects as it moves in a slow directional current through the widened corridors between cells, from the arterial side of the parenchyma toward the paravenous drainage routes that lead, ultimately, out of the skull entirely. The cargo is specific. The molecules are named and studied and chemically understood, and the consequences of leaving them in place, of slowing or obstructing or simply shortening the window during which they are cleared, are among the most significant the field of neuroscience has identified in recent decades. Amyloid beta, in its soluble form, is a molecule of modest size, small enough to drift freely through the interstitial fluid, small enough to be carried in bulk flow, small enough to cross into paravenous drainage channels and exit the brain without incident. In a brain where glymphatic flow is functioning well, the concentration of soluble amyloid beta in the interstitial space remains in equilibrium. Production and clearance are roughly balanced. The molecule is made, it drifts, it is carried away. The fluid moves, the channels are open, the balance holds. Tracer studies in living animals have confirmed this directly. Fluorescently labeled amyloid beta, injected into the interstitial space of a sleeping mouse, moves efficiently through the parenchyma and disappears into paravenous drainage within hours, cleared from the tissue at a rate that corresponds precisely with the rate at which glymphatic flow is moving. What happens when the clearance slows is a question of chemistry and time. Amyloid beta in solution at normal interstitial concentrations remains soluble. It drifts without aggregating. The individual molecules do not interact in ways that change their structure, but solubility is a concentration-dependent property. When clearance is impaired, when the fluid moves more slowly, when the channels are narrower, when the nightly window of peak flow is shortened, the concentration of soluble amyloid beta in the interstitial space rises, slowly, incrementally, over timescales that can span years. And as concentration rises, the chemistry changes, the molecules begin to find each other, they begin to interact. Short oligomeric clusters form, small assemblies of 2 or 3 or a handful of amyloid beta monomers, loosely held together, still soluble, but beginning the long process of structural reorganization that ends somewhere else entirely. In the narrow extracellular space between neurons, in a brain where that clearance has been insufficient for long enough, those oligomers begin to extend. They recruit additional monomers from the surrounding fluid. They elongate into fibrils, thin, rigid structures that propagate by addition, each new monomer adopting the cross-beta sheet configuration of the growing end and locking into place. The fibril grows. It grows slowly, over months and years, in the deep interior of a brain that does not feel it happening, in a tissue where the first detectable plaques can precede any clinical symptom by 15 years or more. And at some point along that slow trajectory, the growing fibrillar mass becomes insoluble. A dense, compact amyloid plaque settled between neurons in the parenchyma, no longer drifting, no longer clearable, no longer part of the interstitial inventory, but a permanent structural feature of the tissue, pressing against the cells around it, disrupting the geometry of the extracellular space, and critically, beginning to narrow the very perivascular channels through which glymphatic flow moves. That last fact is the one that carries the most weight, because the plaque does not simply represent a failure of clearance. It compounds the failure. Amyloid deposits accumulating around perivascular channels press inward on the fluid-filled spaces that cerebrospinal fluid travels through, reducing their diameter, increasing the resistance to flow, slowing the current that would carry away what accumulates next. The drain narrows because the clearance failed, and because the drain has narrowed, the clearance fails further, and what accumulates next accumulates in a system with even less capacity to remove it. This is the cycle, not metaphorical, but structural and chemical and measurable, at the center of Alzheimer's disease pathology, and glymphatic failure is not peripheral to it. Glymphatic failure is the mechanism through which it propagates. In mice bred without functional aquaporin 4 water, channels in their astrocyte end feet, animals in which the molecular infrastructure of glymphatic transport is genetically absent, soluble amyloid-beta accumulates in the interstitial fluid at elevated concentrations, even under normal conditions, even with normal sleep, because the bulk flow that would carry it away cannot operate efficiently without the polarized water channels that drive fluid across the astrocyte end-foot layer. Those animals show accelerated amyloid plaque formation. The connection between the water channel, the flow, and the accumulation is direct and experimentally demonstrable, tracing a single causal line from molecular infrastructure through fluid dynamics to the deposition of the protein that defines the disease. A second molecule moves through the interstitial fluid alongside amyloid beta, less discussed, equally important, and in some ways more consequential for the structural integrity of the neurons it comes from. Tau protein in a healthy neuron is an internal molecule wound along the length of the microtubules inside the axon, holding those slender cylindrical structures stable, maintaining the architecture along which molecular cargo is transported from the cell body to the synapse and back. It belongs inside the cell. Its presence in the extracellular space is a sign of neuronal activity, of normal turnover, of the constant low-level release that accompanies the working brain's operation. Tau in the interstitial fluid is not by itself abnormal. Tau in the interstitial fluid that is not efficiently cleared is a different matter. When glymphatic flow moves well, interstitial tau is carried outward through the paravenous drainage routes at a rate that keeps its extracellular concentration low. The molecule is small enough to enter the bulk flow, soluble enough to be transported, and in its normal unmodified form, not inclined to aggregate. But tau's chemistry is sensitive to its environment. In the extracellular space, away from the molecular machinery that normally regulates it, tau can be modified, phosphate groups added to specific positions along its length, changing its charge, changing its folding, changing the way it interacts with other tau molecules. Hyperphosphorylated tau does not simply drift, it begins to associate with other hyperphosphorylated tau molecules. It begins to form the paired helical filaments, two tau strands wound around each other in a slow right-handed helix that are the ultrastructural components of the neurofibrillary tangles found in Alzheimer's disease tissue. Rodent studies have shown that when glymphatic function is experimentally impaired, interstitial tau concentrations rise, and the probability of tau aggregation in the surrounding tissue increases correspondingly. In human subjects, the picture emerges from a different direction, from epidemiological data linking short habitual sleep duration with elevated tau concentrations in cerebrospinal fluid and plasma, and from biomarker studies showing that even a single night of complete sleep loss produces measurable increases in CSF tau the following morning. The brain that did not clear its tau during sleep carries more of it into the next waking day, and the brain that carries more tau into the next waking day begins the following night with a higher baseline, a slightly less favorable chemistry, a marginally greater probability that the slow process of aggregation will advance. Inside a neuron, where neurofibrillary tangles have formed, the microtubules along which cargo once traveled have lost their stabilizing partner. They begin to disassemble. The axon's transport infrastructure degrades. Mitochondria that should be moving toward energy-hungry synapses stall in place. Synaptic vesicles that should be replenishing the terminal accumulate in the cell body. The synapse, deprived of the supplies it needs, begins to fail. The neuron that owns that synapse begins to withdraw, and the network of which that neuron was a part begins, very slowly, to lose one thread in the weave, one connection, one voice, one point in the pattern. None of this is sudden. None of this is visible from the outside until a great many such losses have compounded over a great many years. What is visible, eventually, is a person who cannot remember what is happening far earlier at the level of the interstitial fluid is a protein drifting where it should have been cleared, finding other proteins like itself, and slowly changing into something that holds on. The brain's waste load is not limited to these two proteins. The tissue that consumes roughly a fifth of the body's total energy budget, despite occupying a small fraction of its weight, generates waste in proportion to that consumption, and the variety of that waste is considerable. Neurons firing continuously produce metabolic debris that accumulates in the extracellular space, oxidized proteins too large to cross the blood-brain barrier and enter the bloodstream, lipid peroxidation products from the high turnover of membrane components in synaptic terminals, cellular fragments from the continuous process of synaptic remodeling that underlies learning and memory. The blood-brain barrier, which protects the brain from pathogens and maintains the chemical purity of the central nervous system, is simultaneously a constraint on the brain's ability to dispose of its own metabolic output. Molecules that would simply diffuse across a capillary wall in peripheral tissue and enter the lymphatic system for processing cannot do the same in the brain. They accumulate in the interstitial fluid. They wait for bulk flow to carry them outward. That waiting has a rhythm. The interstitial space expands during slow-wave sleep. The corridors between cells widen. The resistance to bulk flow drops, and the current that moves through the parenchyma strengthens. In the long hours of the night, the metabolic debris of the preceding waking day begins to clear. Not all at once, not dramatically, but in a steady directional movement of interstitial fluid carrying its dissolved cargo toward the purtuvinous drainage routes that lead out of the tissue and eventually out of the skull. The brain's chemical environment shifts. The interstitial space, which by the end of a long waking day has accumulated the residue of hours of intense activity, slowly returns toward the cleaner baseline from which the next day's operation will begin. Among the molecules caught in that current, two serve as particularly sensitive markers of the chemical state the brain is trying to restore. Lactate, produced in astrocytes as part of the metabolic partnership through which those cells supply neurons with the fuel they need for sustained firing, accumulates in the extracellular space during periods of intense neuronal activity. The astrocyte takes in glucose, processes it, and exports lactate into the interstitial fluid. The neuron takes up that lactate and burns it for energy. The system works well during the waking hours of high demand, but the lactate concentration in the extracellular space after a long and cognitively active day reflects the scale of the demand that has been met, and some of that accumulated lactate is not immediately metabolized. It sits in the interstitial fluid, contributing to a slightly acidic chemical environment, part of the cumulative metabolic load that bulk glymphatic flow is built to reduce. Glutamate, the brain's primary excitatory transmitter, is another molecule whose extracellular concentration the sleeping brain works to normalize. Glutamate is released from synaptic terminals with every excitatory signal, and while astrocytes take up much of what is released, some fraction escapes reuptake and accumulates in the extracellular space during sustained periods of high synaptic activity. In excess, glutamate is not neutral. Prolonged elevation of extracellular glutamate can activate receptors in ways that stress the postsynaptic neurons exposed to it, contributing over time to the kind of metabolic strain that healthy neural tissue is not equipped to sustain indefinitely. The slow movement of interstitial fluid through the parenchyma during sleep carries some of this surplus away from the synaptic zones where it accumulated, diluting it, redistributing it, reducing the concentration at receptor surfaces where it would otherwise remain. The chemical noise that builds through a long active day is in part carried away by the same current that clears the proteins. There is a quality to the interstitial space of a brain well into a waking day that can be described with reasonable accuracy as cluttered. Not visibly, the space is measured in nanometers, invisible not just to the naked eye, but to ordinary light microscopy, but chemically in the sense that the concentration of dissolved molecules in the extracellular fluid reflects the accumulation of hours of metabolic output. Incompletely cleared, gently altering the environment through which neurons must communicate. Lactate at a slightly elevated level, glutamate above its resting baseline, amyloid-beta in its slow continuous accumulation, tau drifting in the space it was released into, oxidized proteins and membrane fragments from the turnover of synaptic structures. The brain is not degraded by this. It continues to function, but there is a chemical distance between a brain beginning a day and a brain ending one, and that distance is the work that sleep must do. Hours into slow-wave sleep, the current runs through widened channels. The clutter in the interstitial space begins to thin. Lactate concentrations measured in sleeping animals show the gradual, steady decline that bulk flow would predict, not the sharp drop of active metabolic consumption, but the slower reduction of a substance being carried away by a current rather than burned in place. Glutamate follows a similar trajectory, elevated at sleep onset, falling across the hours of the night, reaching its lowest concentration in the deep slow-wave sleep that delivers the strongest glymphatic flow. The extracellular environment over the long arc of the night becomes quieter. The chemical state of the tissue shifts toward the cleaner baseline that optimal neuronal function requires upon waking. There is one more molecule in this inventory that carries a particular weight, not because it is a waste product, but because it is both. Apolipoprotein E is a protein that moves through the brain's interstitial fluid as a lipid carrier, a particle that collects cholesterol and other lipid components released during the normal turnover of neuronal membranes, and redistributes those lipids to neurons that need them for structural repair. After intense activity, after synaptic remodeling, after the normal cellular maintenance that every neuron requires to sustain its elaborate structure, lipids are released into the interstitial space and must be collected, packaged, and delivered back to cells that can use them. Apolipoprotein E is the packaging. It moves through the interstitial fluid, collects lipids, and travels toward the neurons that require membrane components for ongoing maintenance. The glymphatic pathway carries it. The same bulk flow that transports amyloid-beta and tau toward paravenous drainage also carries apolipoprotein E particles through the parenchyma, delivering lipid cargo to neurons along the flow path. The current that clears the waste also serves as the delivery route for this molecular maintenance supply. A single fluid movement, driven by arterial pulsation through the periarterial channels and across the astrocyte end-foot layer, performs both functions simultaneously, sweeping debris outward while distributing structural components inward through the tissue. This dual function becomes significant when considered alongside the genetics of Alzheimer's disease. The gene that encodes apolipoprotein E has several known variants, and one of them, the variant designated epsilon 4, is the strongest common genetic risk factor for late-onset Alzheimer's disease, yet identified. Carriers of two copies of this variant have a substantially elevated lifetime risk of developing the disease compared to individuals carrying the more common variants. The reasons are not fully understood. The epsilon 4 variant produces a slightly different form of the protein, differently folded, binding lipids with somewhat altered affinity, clearing more slowly from the interstitial fluid than the more common variants. And this difference in clearance dynamics means that in a brain whose glymphatic flow is also reduced, apolipoprotein E may spend longer in the interstitial space, in higher concentration, in the same fluid that already carries elevated amyloid-beta and tau. Whether impaired glymphatic transport of apolipoprotein E represents a direct link between the genetic variant and elevated disease risk is a question the field is still working through. The evidence is not yet complete. What is clear is that the molecule depends on the same fluid pathway as the proteins whose accumulation defines the disease, that its clearance dynamics are altered by the genetic variant associated with the disease, and that when glymphatic flow is reduced by sleep loss, by aging, by aquaporin 4 depolarization, the concentration of apolipoprotein E in the interstitial space rises alongside the concentrations of amyloid-beta and tau. The same current that fails to carry away one carries less efficiently of all. The brain's interstitial fluid, viewed this way, is not a passive medium. It is an active environment, chemically complex, continuously changing, regulated by the movement of bulk flow through a tissue that works at extraordinary metabolic intensity and generates waste in proportion to that intensity. The extracellular space between neurons is not empty. It holds the chemical history of the waking day, the proteins of synaptic activity, the metabolic residue of energy expenditure, the lipid components of cellular maintenance, and the neurotransmitters of all the signals that have passed through the tissue since the last time the channels widened and the current ran strong. Every waking hour adds to the inventory. Every hour of deep sleep begins to reduce it. There is something that becomes clear when the full inventory is laid out this way, when amyloid beta and tau and lactate and glutamate and apolipoprotein E are understood not as separate concerns, but as components of a single shared chemical load, all present in the same narrow interstitial space, all depending on the same bulk fluid current for their regular clearance or redistribution. The brain does not have separate systems for managing each of these molecules. It has one current. The current moves through the expanded channels of the sleeping brain, carrying everything that is soluble and movable and present in the extracellular fluid, sweeping the full inventory toward the perivenous drainage routes that lead out of the tissue. The specificity of what is cleared is not a feature of a sorting mechanism. It is a feature of what is present. Everything that is dissolved in the interstitial fluid when the current runs is carried. Everything that is too insoluble, too aggregated, too large to be swept is left behind, pressing against the tissue, narrowing the channels, making the next night's clearance a little harder than the last. The soluble amyloid beta cleared efficiently tonight is the plaque not formed tomorrow. The tau carried outward through the perivenous channels during the deep slow hours before dawn is the neurofibrillary tangle that does not begin to grow this year. The lactate and glutamate normalized by bulk flow across the night are the cleaner extracellular environment with which the brain greets the next waking day. These are not large events. They are not felt. They produce no sensation, leave no memory, occur in a tissue too deep and too small to be perceived from the outside. But across years, across decades, across the full span of a life, they add up to something that can be felt, or, When the clearance is insufficient, something whose absence can be felt, slowly, in the growing difficulty of recollection, in the gradual fading of the precision that thinking once had. The same current that sweeps the debris outward delivers the apolypoprotein E inward. The same fluid, the same channels, the same single hydraulic event driven by the pulse of every heartbeat through the periarterial sheaths and across the astrocyte end-foot layer and through the widened extracellular corridors of the sleeping brain. The waste leaves, the maintenance supply arrives, the chemical environment of the tissue, which by the end of a long waking day reflects the full accumulation of hours of metabolic activity, shifts slowly across the night toward the clean, balanced, quiet state from which the next day's operation can begin. And in that quiet, in the slow movement of clear fluid through dark tissue, carrying dissolved proteins and metabolic residue outward while ferrying lipid cargo to the cells that need it, there is a kind of completeness, not the dramatic completeness of a visible event, but the quiet completeness of a system doing what it was built to do, in the dark without witness, over the long unhurried hours of sleep. The molecules that would otherwise accumulate are being carried away. The channels that would otherwise narrow are being used while they are still open. The interstitial space that would otherwise clutter with the residue of waking is very slowly, over the course of the night, being emptied and refreshed. The fluid moves through the deep tissue of the sleeping brain. It carries what the brain has produced. The channels are open, the current runs slow and steady through the widened corridors between cells, and the molecules that drifted all day in the narrow extracellular space, the small soluble proteins, the metabolic byproducts, the lipid carriers doing double duty as delivery vehicles, are moving now, carried in a current too slow and too small to feel, through a darkness too complete to see, toward drainage routes that lead out of the skull and into the cervical lymph nodes, where the brain's internal waste stream meets the body's broader immune system and is finally quietly processed and dispersed. The amyloid-beta that drifted freely through the interstitial fluid this morning will be gone by the time the brain wakes. The tau that accumulated through the afternoon is being carried outward now. The lactate that rose through a long and cognitively demanding day is falling slowly as the current runs. The apolypoprotein E particles are moving through the tissue, collecting lipids, delivering them to neurons that will use them to repair and maintain the membranes they depend on for every signal they will send tomorrow. One fluid current, one slow movement through one dark and ancient tissue, carrying two kinds of cargo in opposite directions at once, the waste out and the maintenance in, the accumulation cleared and the supply delivered, the chemical history of the waking day dissolved into the flow and carried away into the long, quiet, still continuation of the night. In the deep tissue of a brain that has been sleeping for decades, something has been slowly changing. The change is not in a single cell. It is not in a single vessel. It is not in a single vessel. It is in the geometry of the passageways themselves, the fluid-filled sleeves that run alongside every penetrating artery, the narrow corridors between neurons, the small openings through which the brain's internal river has always moved. Those openings have been narrowing for years, the way a stream narrows in a long drought, not all at once, but gradually, measurably, in increments too small to feel, but too consistent to reverse. A perivascular channel in an aging brain, seen in cross-section, looks different from the same structure in a young brain. The arterial wall has stiffened with the slow deposition of proteins in its layers, losing some of the elastic give that once transmitted each heartbeat outward into the surrounding fluid as a gentle rhythmic pressure wave. The astrocyte end feet pressed against the vessel wall have shifted their internal organization over the years, their aquaporin 4 channels no longer quite as densely concentrated at the vessel-facing surface, their polarity softened by the accumulated effects of oxidative stress and cellular age. And around these altered channels, in the tissue beyond them, amyloid-beta peptides that have been produced steadily as a byproduct of normal synaptic activity have begun to deposit along the outer margins of the perivascular space itself. Small clusters at first, then larger ones, pressing slowly inward from the surrounding parenchyma, like sediment accreting against the walls of a narrowing pipe. This is the image the science has assembled, piece by piece over the years since the glymphatic system was first observed. Not a sudden failure, but a slow convergence. A drain that has been working continuously for a lifetime, and that has been narrowing for nearly as long. The brain that is accumulating amyloid beta along its perivascular channels is also, because of that accumulation, becoming less able to clear it. The amyloid deposits alter the geometry of the channels they line. They stiffen the tissue that surrounds the arteries whose pulse drives CSF inward. They change the mechanical properties of the perivascular environment in ways that slow the pressure wave, reduce the inflow, and allow more amyloid beta to remain in the interstitial fluid long enough to precipitate into insoluble form and add to the deposits that are already there. The cycle does not require any additional failure to sustain itself. Each step creates the conditions for the next. The drain narrows, more accumulates, the narrowing continues. In Alzheimer's disease, affecting more than 50 million people across the world at any given time, this convergence is among the most studied biological phenomena of the last 2 decades. The sleep disturbances that accompany Alzheimer's, the fragmented nights, the reduced slow wave depth, the early waking, were long understood as symptoms of the disease, consequences of the neurodegeneration that had already occurred in the circuits governing sleep. What the glymphatic research has altered is the temporal direction of that understanding. Sleep disturbances, it now appears, can precede clinical Alzheimer's symptoms by years, sometimes by decades. They precede the cognitive changes that a patient or family member might first notice. They precede, in some cases, the most dramatic neuroimaging findings. And during those years of subtly disrupted sleep, glymphatic clearance is reduced with every shortened or fragmented night, and interstitial amyloid beta concentrations are, across those same nights, incrementally higher than they would have been with deeper, longer sleep. The perivascular deposits that eventually narrow the channels grew from somewhere. They grew from the interstitial fluid, and the interstitial fluid holds whatever the glymphatic system has not yet carried away. This is not a simple story of cause and effect moving in one direction. The plaques that accumulate also alter astrocyte morphology, changing the shape and the protein distribution of the very cells whose end feet form the walls of the glymphatic conduit. Tau protein, normally wound along the interior scaffolding of neurons, holding the architecture of their long axons in its proper configuration, is released into the interstitial fluid when neurons are damaged or under metabolic stress. And what the glymphatic system does not clear forms aggregates that further damage the neurons from which more tau is then released. The pathology generates the conditions for more pathology. The failure of clearance generates more material that is harder to clear. A single night of poor sleep does not produce Alzheimer's disease. The relationship is cumulative, probabilistic, and shaped by genetic background, vascular health, age, and factors that are still being mapped. But the direction of the relationship, across the evidence accumulated in animal models and in human biomarker studies and in epidemiological data gathered from tens of thousands of people over years, is consistent. Habitual short sleep is associated with elevated amyloid beta in the brain. Sleep deprivation in human subjects, even a single night of it, measurably raises amyloid beta levels in cerebrospinal fluid. And in brains where amyloid beta has already begun to accumulate in perivascular spaces, glymphatic function is measurably reduced compared to brains of similar age without that accumulation. The drain is narrowing. That the drain is narrowing is helping to narrow the drain. Somewhere in the brainstem of a person who will later be diagnosed with Parkinson's disease, the first aggregates of another protein are forming. Alpha-synuclein is a small, normally soluble protein present in the healthy brain in the interstitial fluid and inside neurons near synaptic terminals, where it is thought to play a role in the regulation of neurotransmitter release. Like amyloid beta, it is produced continuously as a part of ordinary brain function. Like amyloid beta, it requires continuous glymphatic clearance from the interstitial fluid to remain at concentrations below the threshold at which it begins to misfold and aggregate. And like amyloid beta, it accumulates when clearance is insufficient. The aggregated form of alpha-synuclein is called a Lewy body, a dense spherical inclusion visible inside the cell body of an affected neuron under fluorescent microscopy, circular and sharply bounded, like a compressed sediment of misfolded protein that the cell can no longer dissolve or disperse. The neurons most vulnerable to Lewy body formation are the dopaminergic cells of the substantia nigra, a small darkly pigmented structure in the brainstem whose neurons extend their axons deep into the forebrain and are essential for the initiation and smooth coordination of movement. When enough of those cells are lost, the motor symptoms of Parkinson's disease emerge: the tremor, the rigidity, the slowing and shortening of movement that define the disease as most people encounter it. But the Lewy bodies that eventually reach the substantia nigra do not always originate there. In many cases, they appear first in lower brainstem structures, in neurons that regulate the body's behavior during REM sleep, among others, years or decades before any motor symptom appears. The first sign that something has gone wrong is sometimes not a tremor of the hand, but a disturbance of sleep. Specifically, a disturbance of the stage of sleep in which the body is normally paralyzed while the dreaming mind is active. In that stage, a healthy person's muscles are held still by the active inhibition of the motor system, a suppression so complete that the most vivid dream produces no visible movement in the sleeping body. In some individuals, years before Parkinson's is diagnosed, that inhibition fails. They move during their dreams, they act them out, they speak and reach and sometimes strike. This condition, REM sleep behavior disorder, is now recognized as one of the earliest detectable signals of Parkinson's pathogenesis. The probability that a person diagnosed with REM sleep behavior disorder will later develop Parkinson's disease, or one of a small number of closely related conditions, is very high, somewhere in the range of 80 to 90% over a decade or more of follow-up in the studies that have tracked these patients. The neurons governing REM-associated motor inhibition are among the first to accumulate Lewy bodies. The REM behavior disorder is, in this understanding, not a symptom that arrives alongside the disease. It is the disease at its earliest observable stage, expressing itself in the one tissue that happens to produce a measurable behavioral sign before the dopaminergic loss has become large enough to affect movement. What connects the glymphatic system to this sequence is the question of what would have happened if alpha-synuclein had been more efficiently cleared during the years before those first aggregates formed. The question is not answerable with the current evidence. The relationship between glymphatic function and Parkinson's pathogenesis is less extensively characterized than its relationship with Alzheimer's disease, and the causal directions are harder to disentangle. But the structural logic is the same. Alpha-synuclein is present in the interstitial fluid. Glymphatic bulk flow carries interstitial fluid through the parenchyma and toward paravenous drainage. Impaired glymphatic clearance, in principle, increases the dwell time of any soluble protein in the interstitial space, including alpha-synuclein, and longer dwell time increases the probability of concentration-dependent aggregation. The sleep disruption produced by the earliest Lewy body accumulation in brainstem circuits could itself impair glymphatic function, reducing slow-wave sleep depth, fragmenting the physiological state in which glymphatic clearance runs fastest, allowing more alpha-synuclein to remain in the interstitial fluid, available to aggregate into more Lewy bodies that impair sleep further. This is the same structural logic as the Alzheimer's cycle, a slowly tightening loop, years in the making, beginning in tissues so deep and so structurally obscure that no clinical sign appears until the loop has been running for a very long time. A head injury leaves no external mark on the brain. The skull absorbs the initial force, or does not, and the brain inside, unattached to the bone that houses it, floating in its fluid cushion, continues moving after the skull has stopped, compressing against the inner surface, rebounding, rotating on its own axis in ways that stretch and shear the long axons of neurons that were never designed to flex in that direction. The damage from a moderate traumatic brain injury is diffuse, mechanical, and immediate. What follows it is chemical. In the days after a brain injury of moderate severity, the perivascular spaces in the affected tissue are compressed by inflammatory edema, fluid accumulating in and around the blood vessel walls as the immune response floods the area with cytokines, and the blood-brain barrier loses some of its selectivity. The astrocyte end feet that normally press close against the vessel wall and form the structural lining of the glymphatic channel swell with the accumulating fluid, their membranes distorted, their aquaporin 4 channels displaced from their normal positions at the vessel-facing surface. The channels themselves, the fluid-filled periarterial sheaths through which CSF enters the brain, parenchyma, are narrowed by the pressure of the surrounding edema. The glymphatic system in the tissue immediately surrounding a moderate brain injury can lose the majority of its functional capacity within the first days of the injury period. Measurements in animal models place the reduction at 60% or more compared to uninjured tissue, and this reduction occurs at precisely the moment when the injured tissue is generating more metabolic waste, more inflammatory cytokines, more debris from damaged and dying cells than it will produce at any other point in its life. The demand on the drain is highest when the drain is most compromised. This acute glymphatic failure following traumatic brain injury is one of the more precisely documented aspects of glymphatic biology, and its implications extend in two directions simultaneously. In the short term, the accumulation of neurotoxic debris and inflammatory mediators in perivascular spaces that cannot drain them efficiently contributes to the secondary injury cascade, the wave of delayed neuronal death and tissue damage that follows the initial mechanical insult and accounts for a significant portion of the final neurological deficit in survivors of moderate to severe injury. In the longer term, for the many athletes and military veterans who sustain repeated injuries over years or decades, the picture has grown more troubling with each successive study. Chronic traumatic encephalopathy, The neurodegenerative condition, documented in athletes who played contact sports and in combat veterans exposed to blast injury, is defined by the accumulation of tau protein in a specific distribution across the brain, concentrated around blood vessels and in the depths of the sulci, the folded furrows of the cortical surface. The perivascular distribution of tau in chronic traumatic encephalopathy is not incidental to the glymphatic system's function. Tau accumulated around blood vessels in a condition driven by repeated brain injury in tissue where aquaporin 4 polarity has been disrupted and perivascular channels have been repeatedly compromised, suggests a connection between impaired glymphatic clearance of tau following each injury and the eventual pathological accumulation that characterizes the condition. The evidence does not yet establish this mechanistically in human tissue with the certainty that would settle the question. But in animal models of repeated mild traumatic injury, AQP4 depolarization persists beyond the acute inflammatory period. Tau accumulates in perivascular locations. The pattern is consistent with a drain that was damaged, repaired partially, damaged again, and over many cycles, lost more of its function than each individual injury would have cost it. There is a quieter version of glymphatic decline that does not begin with an injury or a disease. It begins with a birthday. The change associated with age is so gradual that no single year produces a measurable difference in any given person, and yet across a decade it accumulates to something the data can describe quite plainly. Slow-wave sleep begins shortening in the fourth decade of life in most people. The EEG signature of deep non-REM sleep, the broad, slow waves rolling at less than 4 cycles per second, their amplitude tall and unhurried, loses some of its amplitude first, then some of its duration. The stages of sleep that remain physiologically present grow shallower. The proportion of a night's sleep spent in the deepest, most metabolically active slow-wave state decreases. This change is not pathological in the conventional sense. It is part of the ordinary biological arc of aging, present in virtually every person who has ever been studied in a sleep laboratory across the relevant age range. It is, as a population-level phenomenon, as predictable as the graying of hair or the gradual stiffening of the lens. But the glymphatic system is exquisitely dependent on slow-wave sleep for its peak function. The surge in CSF influx that the research has documented, the expansion of the interstitial space that opens the fluid pathways, the withdrawal of norepinephrine that allows the cellular geometry to shift, all of this is most fully realized during the deepest stages of non-REM sleep. As those stages shorten and shallow across the decades, the nightly maintenance window narrows. The total volume of CSF moved through the parenchyma per night, the total mass of metabolic waste swept toward perivenous drainage decreases. The perivascular channels themselves age alongside the sleep that drives them. The arteries they surround stiffen with the deposition of proteins in their walls and the gradual reduction in the elasticity of the smooth muscle that governs their diameter. A less elastic arterial wall transmits a weaker pressure pulse to the surrounding periarterial fluid. A weaker pressure pulse produces less bulk flow. The mechanical engine that drives glymphatic inflow, the heartbeat, transmitted through the pulsating artery into the fluid sleeve around it, grows incrementally less effective as the vessel wall through which it is transmitted grows incrementally less compliant. In aged rodents, CSF influx rates measured using the same tracer methods that characterize the system in young animals run 40% or more below the rates observed in youth. The same aging brains that have lost glymphatic capacity are also, in many individuals, the brains at highest risk for the conditions whose pathological proteins are precisely the ones that glymphatic flow is responsible for clearing. The convergence is not subtle once it is stated plainly. The brain ages. The brain's waste clearance system ages with it. The materials that the waste clearance system handles, amyloid-beta, tau, alpha-synuclein, are the materials whose accumulation defines the most prevalent neurodegenerative diseases of later life. And the decline of the system that clears them is steepest in the same decades when those diseases most commonly emerge. This is not a coincidence the research has manufactured. It is a relationship the data has uncovered, piece by piece, in study after study, across species and methodologies and research groups that did not set out to find the same thing. A stroke produces its initial damage in seconds. The arterial occlusion, a clot lodging in a vessel too narrow to pass it, cutting off blood flow to the tissue downstream, deprives a region of brain of oxygen and glucose with a completeness that normal cellular metabolism cannot survive for more than a few minutes. The neurons in the core of the ischemic territory begin dying almost immediately. Around that core, in a larger zone of tissue called the penumbra, neurons are under severe metabolic stress but are not yet dead. Their blood supply is reduced but not eliminated. Their membranes are beginning to fail but have not yet crossed the threshold of irreversibility. Whether the cells in the penumbra survive or join the dead tissue of the core is a question of what happens in the hours after the initial occlusion. In the ischemic territory, the arterial pulsation that drives periarterial CSF inflow has stopped. The artery supplying the region is occluded, its wall no longer moving, the pressure wave it would normally generate in the surrounding fluid absent. The glymphatic inflow that depends on that pressure wave stops with it. At the same time, the inflammatory cascade triggered by the ischemic injury floods the perivascular spaces with edema, compresses the channels, and disrupts the aquaporin 4 distribution in the astrocyte end feet surrounding the affected vessels. The post-stroke brain, in the territory of the injury and in the surrounding penumbra, has lost much of its glymphatic function at the moment when it most needs to remove the debris of massive cellular death and the inflammatory mediators driving secondary damage. The debris that cannot be cleared accumulates. Neurotoxic proteins from dying cells enter the interstitial fluid. Inflammatory cytokines build up in concentrations that would not have been reached if the paravenous drainage pathway were functioning. The penumbral neurons, the cells that might survive, given the right conditions, are bathed in a more toxic interstitial environment than they would be if the drain were working, and some of them, as a consequence, do not survive the recovery period. In preclinical research in animal models of ischemic stroke, where the occluded vessel is reopened after a defined period, allowing reperfusion of the ischemic territory, there are experiments in which glymphatic flow in the post-stroke period has been enhanced. The methods used in these experiments are various, some pharmacological, acting on the channels or the fluid dynamics of the system, some behavioral, using the known relationship between sleep state and glymphatic activity to time interventions around the animal's sleep. In the tissue of animals where glymphatic flow was enhanced during the recovery period, the findings have been consistent in their direction. Inflammatory cytokine levels in the peri-infarct zone were lower. The accumulation of neurotoxic debris was reduced. Markers of neuronal survival in the penumbral zone were improved. And in behavioral measures of functional recovery, tests of motor coordination, spatial memory, sensory motor integration, the animals with enhanced post-stroke glymphatic flow performed measurably better than those without the intervention. These are experiments in animals. The translational gap between a rodent model of stroke and a human patient in a hospital bed is real, and no responsible account of this research should ask the listener to overlook it. The mechanisms that work in a mouse brain may not operate with the same efficiency in a human brain. In a patient who is older, who has pre-existing vascular disease, who is receiving other treatments, and whose stroke occurred in a region and at a severity that no animal model exactly replicates. The findings are directional, they are consistent. They suggest that the glymphatic system's recovery after stroke is not a passive process, but one that could, in principle, be modulated, and that modulating it in the direction of increased clearance capacity could reduce the secondary neurological damage that determines much of the functional deficit the patient will carry forward from the event. That is where the science currently stands on this question. Not at a treatment, not at an established clinical intervention, but at a hypothesis with enough mechanistic basis and enough consistent preclinical support to constitute a serious direction for the next generation of stroke research. The brain does not announce what it is doing in the night. It produces no visible sign of the work it performs. From the outside, a sleeping person is still in quiet, and to all appearances, simply absent, gone somewhere that does not generate observable behavior, resting in the simple biological sense of resting. The science that has accumulated over the last decade describes a very different interior. The channels opening, the fluid moving, the waste being gathered from the narrow corridors between cells and carried outward along the perivenous drainage routes toward the cervical lymph nodes, where it will leave the skull entirely and join the body's peripheral circulation. The brain that performs this work every night has been performing it since early life. The channels were present in childhood, running alongside every artery that descended from the cortical surface into deeper tissue. The choroid plexus has been producing cerebrospinal fluid continuously, roughly half a liter every day, in an adult, filling and refilling the spaces around and inside the brain in a cycle that never stops while the person lives. The aquaporin 4 channels, packed into the astrocyte end feet around every brain blood vessel, have been opening and closing in response to the movement of water across the perivascular membrane since the brain first developed the cellular machinery to produce them. None of this began with the discovery of the system. The discovery only gave it a name and gave the people whose brains contained it a reason to think differently about what sleep was doing while it lasted. The relationship between glymphatic function and the diseases that emerge from its failure is, in the current state of the science, one of the most consequential areas of active biological research. Not because it offers easy solutions. The diseases that accumulate amyloid-beta, or tau, or alpha-synuclein in a brain whose glymphatic function has been declining for years are not simply diseases of insufficient sleep, and they will not be simply cured by sleeping longer. The biology is too complex, the pathological cascades too entrenched by the time a clinical diagnosis is made, the individual variation in genetic risk and vascular health and environmental exposure too significant to allow a simple translational pathway from glymphatic biology to disease prevention or treatment. But the relationship is real. Across the evidence in the rodent studies where glymphatic impairment accelerates amyloid plaque formation, in the human biomarker studies where a single night of sleep deprivation raises CSF amyloid beta, in the epidemiological data linking habitual short sleep to elevated dementia risk, in the imaging studies where older brains with worse slow-wave sleep show higher amyloid burden, in the preclinical TBI models where AQP4 depolarization persists and tau accumulates in perivascular locations, in the Parkinson's research where REM sleep behavior disorder marks the earliest visible stage of a disease that began years before in brainstem tissue, where alpha-synuclein was aggregating in the absence of sufficient clearance. Across all of this evidence, gathered through different methods in different laboratories on different continents over more than a decade, the same structural relationship appears. The brain produces waste as a consequence of working. Working brains generate the molecular byproducts of synaptic function, and those byproducts must be removed. The system that removes them runs most effectively in sleep. When sleep is impaired, or when the system ages, or when an injury disrupts its architecture, the waste accumulates. When the waste accumulates, it alters the tissue it accumulates in. When the tissue is altered, the system that was supposed to clear it becomes less able to do so. This is not how every neurodegenerative disease begins, and it is not the only mechanism driving the pathological cascades in the diseases where glymphatic function has been implicated. But it is a mechanism, a physically real, anatomically specific, measurable mechanism operating in every brain, every night, whose gradual failure over years and decades leaves traces that can now be seen in imaging, in biomarkers, in the pathological distribution of the proteins that accumulate where the drain ran slowest. Late in a long life, in a brain that has done its nightly work for decades, the perivascular channels are narrower than they were. The slow-wave sleep that once drove them efficiently has shortened and shallowed through middle age and into later years, each decade's sleep a little less deep than the decade before it, the physiological window for peak glymphatic activity a little narrower each year. The arteries, whose pulse once transmitted efficiently into the surrounding fluid, have stiffened incrementally, their walls less elastic, the pressure wave they generate in the periarterial sheath gentler than it once was. The astrocyte end feet have shifted their aquaporin 4 distribution away from the perfect polarized concentration of youth toward a more diffuse arrangement that allows water to cross the membrane in either direction without the strong directional preference that made the young system efficient. In this brain, the interstitial fluid moves more slowly. The bulk flow that once carried amyloid-beta and tau and the metabolic debris of a day's synaptic activity efficiently toward the paravenous drainage routes moves at a fraction of its earlier rate. What remains behind in the interstitial space has more time to aggregate, to cluster, to precipitate out of solution and into the insoluble forms that the drain could once have carried away, but can no longer reach. The person whose brain this is does not feel the change. The change is not in any sensation they can access. It is in the width of a perivascular channel, in the distribution of molecular channels on an astrocyte's surface, in the amplitude of a slow wave on a recording made in a sleep laboratory that they may never have visited. It is in measurements that require the most sensitive instruments neuroscience has yet developed to make, in fluids that must be collected by lumbar puncture or detected through the skull by isotopes that emit faint radiation. The brain announces nothing of this to the mind it houses. It continues, through the narrowing years, to do the work it has always done, less efficiently than before, without complaint. Every brain that has ever lived has done this work. The glymphatic channels ran in the brains of people whose names are not remembered, in the long centuries before anyone knew what cerebrospinal fluid was, before the choroid plexus had been named, before the astrocyte was distinguished from the other cells of the nervous tissue under the first microscopes, powerful enough to see individual cells at all. The fluid moved through the periarterial sheaths of every brain that slept, in every night of every life, carrying away what needed to be carried, maintaining the narrow chemical balance that kept the tissue alive and working. No one knew it was happening. It happened anyway. The slow waves that marked the deepest sleep rolled across those brains in the dark, hour after hour, their rhythm older than the name given to the process, older than the understanding that sleep was necessary, older than the tools that eventually made the fluid's movement visible. The glymphatic system is not a discovery that created something new. It is a discovery that named something ancient, something that was already present in the first mammalian brains that ever slept, in the evolutionary lineage that had already, by the time the name existed to give it, been refining the system for tens of millions of years. The fluid moves now, in the same way, in the same channels, through the same molecular machinery as it has always moved.